Okay, I think I've clicked all the things I needed to click. Welcome back, everyone. And we start as always with a little prayer. In the name of the Father, the Son, Holy Spirit. Come, Holy Spirit, fill the hearts of your faithful and kindle in us the fire of your divine love. We pray especially for all those who are in harm's way and the victims of the coronavirus, all the infections and dangers that are out there. Sometimes we are oblivious to because of all the well-being that we enjoy in this country, in this nation. As we enter this Lenten season, dear Lord, we ask the gift of your Holy Spirit to inspire us to change what needs to be changed in our lives for the better, and always to help others to do so also as we seek to come ever closer to you, to your gospel, to your truth. In the name of Christ, our Lord. Father, Son, Holy Spirit. Okay, so first of all, we've had 2 weeks of no lectures. So is there any questions or comments from previous stuff? Well, all right, we're moving along. I think we only have 2 more lectures for the beginning of life issues, and then we'll move into end of life. All right. Okay, so I want to look at stem cell research and human cloning today. And to me, this is one of those classical examples of that little saying attributed to Thomas Aquinas, seldom affirm, how do you put it? Seldom affirm, never deny, and always distinguish, making distinctions. All right, so a lot of moral theology and a lot of bioethics is making distinctions. What do we mean? What is going on? Seldom affirm, never deny, and always distinguish. It was attributed to him anyway. And he's considered, Aquinas, the Dominican friar from the Middle Ages, 12th, 13th century, to be the great systematizer of Catholic theology, right? He wrote his famous Summa, his famous theological books, which were the textbooks for the seminary at the time. Italian fellow. At any rate, let's see. So we could ask a question like, is the Catholic Church in favor or against stem cell research? Okay, and if I ask the question just that way, how many of you would say that the Catholic Church is in favor of stem cell research? 1, okay, and how many would say that the Catholic Church is against stem cell research? Okay, none, okay, and then how many are not sure according to how the question is asked, right? That's the thing, we really need to be more specific, okay, because I can tell you if the question is asked that way, we can actually say the Catholic Church is not only not against stem cell research, the Catholic Church is actually involved in stem cell research. It happens to be adult stem cell research or cord blood stem cell research, right? Okay, so tell a little anecdote when I was getting into, so before going to Purdue, before actually going into a graduate program in genetics, I had to prepare for a year because I was in my 50s and it had been at least 10, 15 years since I had been away from formal studies like that. And so for 1 year, I went to Boston and took some courses and also was preparing to take the GREs. Again, I had taken the GREs for about 20 years or more. I guess in the 70s was when I took the GRE. So it had been 30 years. Anyway, I spent a year in Boston preparing, and part of the preparation was I did some research at this the Catholic hospital in Boston. Boston is supposed to be a very Catholic archdiocese. It has a cardinal see and all that. Anyway, St. Elizabeth Medical Center, after Elizabeth Seton, I guess, is a teaching hospital for Tufts University. which the medical center is downtown there. And they have a building, the hospital has a separate building next to it, which is all research. And that building also does all the lab work for the hospital. So all the hematology and everything is done there. It's very convenient. And they do research. So I was doing research there. And that's where I met a number of these Indian postdocs. They come here to the US to get some publications for a few years. And then they go back to India. And if they have those good publications, it gets a better job back in India, typically with the government in some research area. At any rate, one day I was talking to a couple who had gone to give birth at the hospital. And they said, Father, we were bewildered because we knew that we could save the stem cells from the umbilical cord of our baby, okay? But we asked the nurse here at St. Elizabeth Medical Center, in other words, the Catholic hospital, if they could please save the cord, the umbilical cord for us, or the cord blood, right? So that we can save for stem cells. And the nurse said to us, oh, she was sorry, but they wouldn't do that there because that was a Catholic hospital. Okay, so the misinformation was at the level of the staff, of the actual personnel in the hospital. And I said to myself, how sad, because here's a young couple who was willing to go through the expense of saving the stem cells and everything else for future benefit to that child or siblings or even the parents because of the compatibility. And yet that opportunity was lost, was wasted, because of misinformation on the ethical side from this nurse, right? So that prompted me to get deeper into all this study of the stem cells. Plus, in the early 2000s, this topic was really hot, and it was kind of parallel with Dolly, because I think Dolly was cloned in 1995. We'll talk about cloning, mammalian cloning and human cloning in the second half after the break. But it was right around that time that it was in the news, and also Yamanaka with the induced pluripotent cells and so forth. Okay, so that's what we're going to get into today. All right, so this is kind of a summary here with regards to today's topics. We're going to talk about stem cell research, the distinction between human and non-human, then the distinction between adult and embryonic, a couple of other examples of stem cell research that are the cord blood from the umbilical cord, and then also the iPS cells from Yamanaka in Japan. And then with regards to cloning, otherwise known as somatic cell nuclear transfer, that's just a scientific name for the same. It's the phrase is actually describing what's happening. And then for the purpose of reproduction or the purpose of therapy, okay? So this is kind of a summary of what we're going to get into. Again, if there are any questions or comments along the way, just jump in, all right? Moving forward, is that clear enough and the point is large enough? People can see, right? All right, so first, the distinction that we have to make with regards to stem cell research, right? Stem cell research is it human or non-human? Because again, reasonably, it stands to reason that there is some research that we may do. Not only can we do it, but we may do it in animals that we may not do in humans, or at least the trials that are done are sequential, right? So let's say hypothetically, when there's genetic research going on, we start basically with bacteria, okay? And it's also very practical because within 12 hours, more or less, you get an explosion of bacteria from a single transformation, genetic transformation, to billions of bacteria, trillions. So you can gather quite a large amount of DNA just overnight incubating bacteria. So typically, we start with bacteria, and if whatever the scientists are looking for seems to be successful, the sequence has been inserted in the right place and so forth, then they may bump it up to Drosophila, for example, the fruit fly. All right, and again, within a couple of days, we get a population of fruit flies that is pretty significant in numbers, and we'll do the stats for it. If that is successful, then eventually we'll go maybe into fish or something like that. And eventually would have to go to a mammalian model. I'm thinking of a clinical trial, let's say to find an antidote, to find a medication to be applied for humans, right? And so it's been bumped up the scale, if you will, so that if there is no success at one level, then one stays at that level until there is success, and then it moves forward to the next level. Eventually, we have to land into the mammalian model, a model meaning an animal or a plant that is being used in the lab for research, which means that that animal or plant at some point is going to be euthanized or sacrificed for the sake of the experiment, right, for getting the data. And so the one who's going to get it, the mammal who's going to get it typically is Mus musculus, or the mouse. All right, the lab mouse, which many research labs, they raise these mice typically in the basement of the research building. And I can tell you there's very strict protocols for maintaining these mice that is regulated by the government. And if these protocols are violated, the government can close not only the breeding space for the mice, but can shut down the whole lab for a period of time for violation of laws, of regulations. For example, because at Purdue, we were raising mice down at the bottom. It's interesting that the science building in Purdue, well, there are about 4 or 5 science buildings actually in Purdue, it's huge. But the one building that I was at in basic biology had 4 stories above ground and 3 stories below ground. So they actually had 7 stories, okay? The 3rd floor below ground, they had all the electron microscopes there. So the building was in an L shape. The floor plan of the building was huge. It had maybe, I would say about between 30 and 40 labs in each floor, okay? And the L shape was for Eli Lilly, as in Lilly medication, Lilly drugs, all right? Because Eli Lilly was from Indiana, and he donated part of his millions to research there as one of the first biology labs at Purdue. And so all the electron microscopes were down in the basement where there is less vibration, supposedly. But then another floor was for rearing of the mice and also rabbits. And one time I saw an article just in passing that someone discovered that at night, the male mice are singing. They sing throughout the night. They spend the night singing, courting the females. But it's such a high pitch that it's like those dog whistles. The dog can hear it, but we can't hear it, okay? And someone somehow, maybe he had some kind of instrument, and the thing was recording like crazy, and it turns out that all the male mice down there are singing to the females all night long. And no one had ever discovered the quandaries of, anyway. But with regards to regulation, as soon as the female drops the litter, for example, which can be 3, 4, 5, 6 little baby mice, okay, the male has to be taken out right away because the male will start eating the babies, you know. And so that means that typically the graduate student has to be pending, has to be al tanto, how do you say al tanto? Aware, attentive to when the female is going to drop her litter and has to go to the lab at whatever time of day or night to take the male out. Because otherwise it would be in violation of the regulations and that can be shut down. So I'm just trying to say that there are a lot of regulations in place to treat these animals humanely, okay? But at the end of the day, they need to be sacrificed for the sake of the experiment, because obviously we're going to sacrifice mice and and rabbits, and even in extreme case, the chimpanzee who was raised in the lab or in a zoo situation before we sacrifice a human or we test on the human, right? So we need that degree of assurance, which should be the high standard, at least the 99% or the 99.9% p-value. All right, so these are levels that are in place, but at any rate, back to this distinction of with regards to stem cell research, are we talking about human or non-human research? Okay. For example, in the non-human research, there is embryonic stem cell research going on with mice or rats. In this case, typically it's more rats simply because the animal is physically larger and it's easier to observe and to manipulate a spinal cord injury that they're trying to heal with stem cells, with embryonic stem cells coming from the same mother of those stem cells, so that at least there's a 50% genetic compatibility. And so the animal is injured on purpose. The spinal cord is partially severed on purpose to try to see if the stem cells are going to work or not. Okay, that's research that goes on with rodents to try to heal. The hope is to heal, for example, injuries from soldiers or from people who have had accidents in the spinal cord and try to regenerate that site of injury. Okay, so that's the first level that we have to distinguish bioethically, whereby some research may be acceptable in non-human, in animals, but not acceptable in humans. Okay, so that's, primates at all? I'm sorry? Primates? Less and less. It's getting harder and harder to justify research on primates, but and I can't tell you specifically what lab is going on, but it's just my intuition that for very serious things or very high risk, they would test on if it's needed. That's the other thing too. If it's actually needed, a primate as a model, or if a rodent will do. Okay, because of the percentage of genome. So again, kind of thinking out loud here, it may come down to what region of the genome is being tested, because if that region of the human exists in the rodent, then we don't have to test on the primate. But if the compatibility is only with the primate, of which we have like 99%, right, then I guess the chimpanzee will have to be tested.
I think before going to the primates, we'll try with the models,
Yeah, rhesus, for example, which that's where we get the RH factor. Actually, that came from there, from the rhesus monkey, which is RHESUS, is the genus name of the monkey. Yeah. All right. So the whole point here is that we make a distinction. We make a bioethical distinction between human and non-human stem cell research. That's the first one. Okay. So then we're saying that we're talking about human stem cell research. Okay. That leads us to the next level of research, which is now the most obvious one for the ethics of it, is between embryonic or adults. All right, so this is the second level of distinction. Are we talking about embryonic or adult? So as to the question of the professor, is the Catholic Church for or against stem cell research? The student should ask, well, what do you mean, professor? Do you mean embryonic research? First, you mean human and non-human? Okay, human. And then do you mean embryonic or adult stem cell research, right? And those are the levels of distinction. And that's where we can get now into the nitty-gritty of it. And we can say here that whereas the church is against human embryonic stem cell research, the church is actually in favor of human adult stem cell research, okay? And that is based quite simply on the source of the stem cells. The source of the stem cells, which in the case of the embryonic involves the killing of the human embryo. In case of the adult, it involves just consent from the adult because we all have stem cells. Okay, so we're gonna go there. First, let's go to the embryonic. And we have to go back to the beginning of the course when we were talking about that embryonic development, the early embryonic development, and that third stage of development. First, we have the zygote, right, which is a fertilized egg, the new individual. The zygote grows into the morula by simple mitosis, cell division, and eventually the morula will grow into the blastocyst, meaning that the morula will start hollowing out on the inside and will develop two layers. It's the first time that we have two layers of cell differentiation. The outer layer, which is known as the trophoblast, and the inner layer, which is a mass of cells, okay? And so just unimaginatively, it's called the inner cell mass, ICM. For once, there's an acronym that kind of makes sense, it's intuitive. Inner cell mass, ICM. Also, sometimes you see it as the embryoblast. These are the embryonic stem cells. This is what becomes the embryonic stem cells. So you see that physically, yeah, inner cell mass, the ICM, these are the ones that become the embryonic stem cells. So to obtain these embryonic stem cells, necessarily this blastocyst has to be disaggregated, has to be taken apart, right? To get inside and get those ICM out into the Petri dish. So by disaggregating the blastocyst, I am actually killing the blastocyst. That blastocyst is not going to survive that process. And remember that the ICM is what becomes the embryo proper during the development. And that's why they are the theoretical gold standard. Yes, they are the gold standard because these cells are pluripotent. Okay, sorry, pluripotent. And we'll get into potency in a moment. But just to be clear that the embryonic stem cells of whatever species come from the ICM, all right, from the inner cell mass. And to do so, well, that blastocyst needs to be disaggregated, meaning that that blastocyst will die, has been killed, of whatever species. So let's look at potency for a moment. By way of just a general background, there are 4 levels in general of potency, all right? But 2 are kind of absolute levels and the others are a range. And you'll see what I mean. We start with pluripotent cells, which again, this is a little added value of the Latin. Okay, potency is the capacity to develop further, basically we can define it that way. So pluripotent, as the word says, these cells have the capacity to become the entire embryo or to become the entire human. Obviously the zygote, which I think you're familiar with this photo by now, it's actually an electron micrograph. These are photographs. You can see the 2 pronuclei beginning to fuse here, right? So this is a zygote that is beginning syngamy. You can even see the polar body here, polar body embedded in the corona, the zona pellucida, sorry, and then the corona on the outside. So this is the zygote, which is pluripotent because it's the only cell there is. So obviously the whole embryo will come from there, okay? So that was pretty intuitive. But it turns out that the morula is also pluripotent. I don't know if you remember, but I mentioned that each cell of the morula is called a blastomere, just technical blastomere, okay? But each one of those blastomeres is also pluripotent, all right? And that's also why the morula just looks, even to the simple eye, that all the cells look alike, even morphologically the same size and whatnot. There is compaction going on here so that the cytoplasm is being redistributed into smaller cells, but they all have the full genome, obviously, but they all have the potential to become an entire new individual. And this goes back to experiments that were done last century, the beginning of last century, with that German fellow Hans Spemann. He was working with amphibians, with frog embryos and frog moruli. And he would put this moruli on a petri dish and disaggregate the blastomeres of the morula. And for each blastomere, a new little frog would eventually develop. So each frog, each blastomere kind of reverted to being a zygote functionally. You see, because, so here's the morula. Let's say this is at the 16 cell stage. I don't know, maybe it's 34, 32, but somewhere in there, you know, it's certainly more than 8, less than 60. So it's either at the 16 or the 30 cell stage, right, when it's duplicating. But if I pluck out just one of them and put that cell in a Petri dish with the same medium, all right, under sterile conditions and so forth, at the proper temperature, that one blastomere will eventually start within 24 hours, typically will start cell division, mitosis, and split into 2, and then 4 and 8 and 16, and we're back to a morula again for each blastomere. Okay, and if we continue to allow those morulae to continue growing, they will go into blastocyst or blastulus in the case of amphibians, and eventually a little tadpole will come from there. And then the tadpole will develop further into the adult frog and start jumping around, and that frog can reproduce. So that's the proof of principle that the blastomere was indeed pluripotent, because that little blastomere developed all the way up to the adult, who was a fertile adult. Okay, so that's the proof of principle. So that means that even the morula is pluripotent, but then by the time we get to the blastocyst, these cells have lost their totipotency, and now they're pluripotent. Okay, so pluri for many, which multi is also for many. So here is, we see a paucity of our language and a richness on the Latin, in that technically there's a difference between pluri and multi, whereas pluri has more potential than multi. All right, and you'll see why in a minute. So let's look at the blastocyst again. How many cell layers do we have? It's the first level of differentiation, right? So the minimal differentiation that we can have for differentiation is 2. In other words, 2 different types of cells that are going to have 2 different functions that will become 2 different organs or systems, if you will. All right. So again, think functionally. Remember the trophoblast, the outer layer, trophoblast, that becomes what? Placenta, right? Because remember, this is what is in contact physically. Remember, this is a very mechanical thing. All right. The trophoblast is the one that is in contact, direct contact with the endometrium of the uterus, and it's the one that is embedding into the endometrium. So that direct physical contact is going to establish the placenta. So the trophoblast is what becomes the placenta. Whereas the ICM, been mentioning, is what becomes the embryo proper. It's the one that goes through gastrulation and the 3 derm layers, the ectoderm, mesoderm, and endoderm, right? And so it is the embryoblast, or the ICM, the ICM, that becomes the embryo, properly speaking. So we have, at this level of 2 layers of cell differentiation, this is the first time of cellular differentiation, right? Now, it means that the trophoblast has lost the capacity to become the embryo proper, and the ICM has lost the capacity to become the placenta, properly speaking. Okay, so now what is happening is these cells are becoming more and more defined into a particular track of differentiation, or of cellular differentiation, which is a whole process of development at the cellular level, is differentiation. And the goal is to get to fully differentiated cells, which is the vast majority of our cells in the body. Fully differentiated means that functionally they have a specific function. So that liver cells do liver things, and brain cells do brain things, and so forth, okay? So this passage here from totipotent to pluripotent is the first level of differentiation, okay? Then there's a further level of differentiation that will occur, and I'm only focusing on the embryo proper, okay? Which is that ICM. That ICM will become the gastrula, And the gastrula will establish the bilateral symmetry, elongates and starts mimicking things left and right of the body generally for bilateral organs and everything else that is going on. So what do we have? We have the next level of differentiation precisely through the process that is known as gastrulation, okay? It's a very active, dynamic, and fast process of cellular differentiation that is occurring in the embryo. I'm talking here typically like the second week, because in the first week we have implantation, number 5 to 7 days for the little embryo to travel from the ampulla to the endometrium. Now the embryo is implanted and it continues that fast development. Second week is gastrulation, and the main event of gastrulation is to establish these 3 germ layers. Germ layers, ectoderm, mesoderm, and endoderm. Again, the Latin wording, let me bring it a little higher so it can be seen. Can you see? Oh, sorry. Right. And ecto on the outside, meso middle, and endo on the inside. Derm, just tissue in general, not just skin, but tissue. All right. So that the actual skin comes from the ectoderm, for example. Whereas the skeleton, for example, comes from the endoderm, and then the mesoderm, muscle, and many other organs and glands and so forth in the middle of the body. Okay, so this is a general reference to more or less where the organs or tissues are going to be located. The interesting thing about the ectoderm, though, I always find fascinating, is that the nervous system also comes from the ectoderm. It's very interesting because if you think about it anatomically, physiologically, the nervous system is the innermost system of our body, especially the central nervous system. Consider, to get to the brain, physiologically, we have to go first through scalp, we have to go through skin, then we go through layers of muscle, then we go through skull, then we go through meninges and membranes, and finally we get to the neural tissue of the brain. So all those layers, okay, even though they're thin, they're definitely there. And so, and the same argument holds when you go down the brain into the spinal cord. It's literally inside the vertebrae, inside the skeletal system. And so in a very real anatomical, physiological way, the nervous system is the innermost system of our bodies. And yet it doesn't come from the endoderm or even the mesoderm, it actually comes from the ectoderm. It's a process that is known as invagination. The ectoderm pours into that middle groove, right? That occurs during gastrulation. Very fascinating process, that is very fast. But imagine all of the regulatory mechanisms that have to be in place and clicking at the right time for that to happen properly, to develop eventually over the years an entire central nervous system, all the way from the brain and all its complexity with the gray matter, the white matter, the convolutions, the sulci and all that, and down the spinal cord, and then the nervature that will go out, the ganglia and everything that will branch out to every part of our body. Even all the way out to the sensory nerves, which are not just tact, but also cold and heat, that are different types of nerve endings, all the way back out to the surface of our body, which is the skin, right? The integumentary system. So all this begins during gastrulation. Ecto, meso, and endo? Okay, so meso and endo. All right, so this is a reference to generally where those tissues or organs are going to be located in the body, generally. So that the ectoderm is going to be the outside of the body, typically the skin, with all its complexity, all the glands and everything else that are there, but also the nervous system that comes from the ectoderm, just a biological curiosity, if you will. The mesoderm are going to be the organs in the middle, typically the digestive tract and many glands that we have, muscle. And then the endoderm, typically it is the skeletal system, way into the body. And also there is a mixture, many organs have a mixture of mesoderm and endoderm, okay? And it gets pretty complicated. It's not a course in anatomy or physiology, but generally these three layers establish the formation of our organs. All of the organs and tissues, I would say the takeaway here is that all of the organs and tissues of our body come from these three cell layers, these three tissue layers, okay? And that's why they're known as germ cells, germ layers. But by the time we get to this layer, then the embryo is now, has gone into, this is still pluripotent, now moves to multipotent as the gastrula continues to develop, and the embryo makes a transition from embryo to fetus. Remember, at the 8th week, more or less, when all the organs are formed. Now we're down to multipotent cells, okay? Now, multipotent cells are within a particular organ or system. So that, for example, the circulatory system will have multipotent cells, okay? That means that those cells typically are predisposed to making only circulatory system cells. For example, bone marrow. Bone marrow is the one that makes the white blood cells, the red blood cells, right? And so, I don't know, are platelets made in the bone marrow also? Platelets? So
Everything is.
all the circulatory system comes from the. Okay, so that to me is a clear example of multipotency, right? Multipotency is the level of organ system. So that generally and naturally, let's say circulatory precursors will not become neuronal cells. And neuronal precursors, of which we have, for example, in the hippocampus of the brain, right, will not become muscular cells and so forth, okay?
What's the Latin you were referring to before, the difference between multi and pluri? Pluri being many, and then what is multi?
Exactly, that's why I say there is a paucity, there is a poorness of the language in English, because we will say many for both. But in Latin, they make a difference between pluri and multi, whereby pluri is broader than multi. So for example, the cells of the blastocyst are pluripotent, okay? And that's the gold standard is there, in that the ICM is pluripotent, but let's say mesenchymal cells or cells from the bone marrow are multipotent. They have lost their pluripotentiality. So what does that mean functionally for the researcher in the lab? It means that from multipotent cells, let's say bone marrow, that researcher normally can only get circulatory cells, cells in the circulatory system, okay? Whereas with an ICM, that researcher could potentially develop many of the 220, more or less 220 cell lines that we have in the body. And that's why, again, the pluripotent cells are the gold standard for the industry, for stem cell research, because potentially we could get all of the cell lines, or at least most of the cell lines that make up our body, which is considered about 200 something cell lines that we have in our body, okay? Whereas when the cell is already multipotent, it has already lost some of the broader potentiality. It's already narrowed down into a particular organ system, right? Whether it's digestive system, circulatory system, muscular system, nervous system, immune system, you see? So multipotent has lost potentiality functionally. That's the whole point there. And again, that's why we go back. It's kind of a circular argument. The researchers want the gold standard because since the whole embryo comes out of the ICM, then that is the ideal stem cells to work with for developing at least most of the cell lines that make up our body. Of course, what's the background to this? I failed to mention it, should be pretty obvious by now, is regenerative medicine, right? To heal injuries or diseased organs or injured organs that are no longer functioning well, to heal them. Talking about pancreas, liver, you name it. Spinal cord, all right? Maggie?
Could you just, I'm sorry, could you define a cell line? I'm not sure what that means.
Sure. Okay, how do we define cell line? Cell lines are precursors to all the differentiated cells in the body. Let's hash that out a little bit. So cell lines are precursors to all the differentiated cells in our body. Let me talk briefly about fully differentiated cells, which will help put it in context. The vast majority of our cells, okay, so we have trillions of cells in our body, right? Even a baby has trillions of cells. And when we get to adults, maybe we have quadrillions of cells. But the point is that we are just invaded with a huge amount of cells, the vast majority of which are alive. And not only are they alive, they're alive functionally, they're doing their thing. So the brain cells are doing brain things, and lung cells are doing respiration, whatever the function, think functionally. So fully differentiated cells means that these cells are doing their thing. They're metabolizing in their particular organ, tissue, all right? And doing their thing and not another cell's thing. So for example, lung cells in the alveoli, they're doing the gas exchange, they're not thinking, they're not doing neuronal things, and so forth. We can make that argument for every cell of our body, for the vast majority of cells. They are in their particular function. That's what we call fully differentiated cells, okay? And that's the general trajectory of all the cells in the body from the time we begin as a single cell to the time we're born in trillions of cells and continue to grow into adulthood so that the body can function as a whole, what we call metabolism. All right, so those are fully differentiated cells. Now, it turns out that these fully differentiated cells, like the immediate precursors of them, are about 200 cell lines, okay? About 200 cell lines. So for example, let's say what makes up, let's stay with stem cells. Let's stay with the bone marrow, because bone marrow is a very good example. Bone marrow, we can say that bone marrow, all right, is a precursor, it's a cell line for white blood cells, red blood cells, platelets. These are three different cell types that we have in our blood. And whereas they're all in the blood, okay, they belong to the circulatory system, they have very different functions. So that RBCs, red blood cells, they are involved in the gas exchange. They pick up oxygen in the lungs, take it to the tissue, they pick up CO2 in the tissue, take it to the lungs, all right? The white blood cells, and there are different types of white blood cells, they're involved in the immune system. They're not involved in gas exchange, but they're in the circulatory system, okay? So those are two different cell lines there. Platelets. Platelets are not involved in the gas exchange, they're not involved in the immune system directly, but they're involved in making a scab, all right, when there is an injury. And so those are three different lines that you see there. And like that, another 200 make all the different differentiated cells or tissues that we have in our body. So those are cell lines. And the idea is, at least when this started with this whole idea of stem cell research, was to develop in the lab at least the vast majority of those cell lines that would be immortal, all right? In other words, that would be available for healing eventually when that injury happened. Let's say in diabetes, for example, there would be islet cells from the pancreas develop the cell line for pancreatic cells as a cell line to be available for diabetic people who could then, with that cell line, with those stem cells, they could heal their diabetes because those cells would replace the disease or the injured pancreas. All right, so that's a cell line. The idea was to use these cell lines as healers, how do you say that, as replacement for the defective cell lines that a person would have in their body, either because of an injury or because of disease or because of some genetic abnormality. So cell lines were supposed to replace or to heal the non-functioning lines that a person would have, that a patient would have in their body.
They are the actual breeders of cells.
That's a good way of putting it. Yeah, the breeder. Hmm? Yes, they are like the germ cells, okay? It's like the precursor. For example, the precursors of the eggs and the sperm, these are germ cells that are embedded in the ovaries and in the testicles, and they are the germ cells that produce those what eventually become differentiated cells, like egg and sperm, because those cells are differentiated, very differentiated. They have very specific functions, right? But they come from precursors, the breeders of those cells.
So then, do bodies have the same cell line, or is it
Yes.
our own body?
Okay, so we have generally the same line, the same cell lines as one species, but then each one of our cell lines is also fingerprinted, if you will, by the specific genome, by the specific genetics of our own individual, all right? So generally we have the same cell lines, but there are specifics that are details in the genetic sequence that are specific to each one of us. So it's like a genetic fingerprinting. All humans have fingerprints, but each fingerprint is somewhat different, right? So it's at the level of general or at the level of detail.
So is the bone marrow the cell line, or is the platelets the cell line? The one that comes from the bone marrow.
It's the bone marrow, right? The bone marrow is a cell line.
Platelets are net cell, fragments of cell. They don't reproduce.
Platelets are a type of cell. So you would have a white blood cell, you would have a blood cell, a platelet, you would have,
So it's the source of all of those.
so.
Yeah. Yeah, in other words, the platelet is a fully differentiated cell. It's a dead end. In other words, it has fully differentiated or it has a specific function. You can think of differentiated as a specific function and no other function, okay? So differentiated cells come from cell lines. They come from cell lines, okay? All right, so again, this is just so that we understand why the ICM is that gold standard for the stem cell research field, because the whole embryo comes from there, human and non-human, for any species, okay? A fly, I don't care. The whole embryo comes from there. Okay, so, but I keep saying it's a theoretical gold standard. Theoretical gold standard because, like always in nature, there's a glitch. All right, let's see if the glitch is here. Yeah, okay, so I need to talk about the glitch. This is the glitch. Let's go to the lab. Because one thing is the theory, and another thing is the practice, right? Let's go to the lab. When these embryonic stem cells are, when the ICM is taken out of a blastocyst and put into a petri dish and turned into embryonic stem cells, all right? And then these embryonic stem cells are injected into a site of injury. I'm not talking about human, because here is where the clinical trials are important. This has been done typically in mice or rats, like I mentioned, okay? Let's take a female rat and take out some eggs, fertilize them from a male rat, obviously, grow the little embryo to the blastocyst stage, take that ICM, grow the cell line in a petri dish. So now we got non-human, we got rat embryonic stem cells. We're following the research, following the experiment. Now we got rat embryonic stem cells. Now we go back to the mother, to the female from where the eggs came, which there's a 50%, at least 50% compatibility, right? Because the egg was from her. We take that female rat and sever the spinal cord partially sever the spinal cord, and then inject directly the embryonic stem cells right into the site of injury to see if that spinal cord is going to regenerate. Theoretically, it should, right? Because the whole rat comes from that ICM. And so theoretically, that's the whole point. It would regenerate the site of injury. And how do we test that? Well, once the rat is severed the spinal cord, not totally, to not kill the rat and to not make her quadriplegic, but paraplegic. In other words, they sever partially the cord so that she becomes paralyzed from the waist down. And literally, she'll drag herself with the forelimbs, and she's dragging her body, the rest of the body, because the rest of the body is immobile from the neck, from the waist down. All right, so she cannot move her hind leg, she cannot move her tail, but she's alive. Okay, she may not even be hurting, by the way, because that side may not cause pain. Well, the injury itself is done under anesthesia, et cetera, it's a surgery. But she's paraplegic, right? So she cannot move her hind legs or her tail. And so it's a drastic injury, but very graphic, can be quantitative, right? Can be, get a lot of data from there. So the embryonic stem cells are injected into her site of injury and given some time for healing and hopefully regeneration. What's the typical result? The typical result is not only is the spinal cord not regenerated, typically she starts growing tumors. She grows actual tumors on the site of injury from the embryonic stem cells. Why? Precisely because those cells are pluripotent and not multipotent. In other words, those embryonic stem cells do not belong, are not already into the nervous system line. They're pluripotent. So you can think that basically the whole embryo wants to grow there. The whole embryo wants to grow there because they're pluripotent. And so she ends up with tumors, which we call cancer. Okay? And that's the typical result when these embryonic stem cells were injected into the site of injury. First, I forgot to mention, first there is an immune rejection, because whereas 50% of that genome is compatible with her own because it's her egg, okay? The other 50% being from the sperm is not compatible. And so there's an immune rejection. The body's ambiguous whether these cells are actually friend or foe, kind of similar to the embryo. But unlike the embryo, when the normal embryo is implanting, that the embryo literally convinces the endometrium to take him in or take her in, right? In the biochemical elegance that goes in, the dialogue that goes on there, these embryonic stem cells don't have the capacity to do that dialogue and convince the site of injury that they're friend and not foe. So the body mounts an immune reaction. So now the rat has to be put on immunosuppressants. Okay, similar, analogous to a person getting a transplant. All right, the rat has to be put on immunosuppressants to tone down her immune system, not shut down the immune system, but just tone it down, like people with transplants. Okay, they take medication to tone down their immune system, immunosuppressants. It just makes them more susceptible. What for us would be a common cold, for them could turn into a pneumonia. All right. So again, think functionally, I translate hypothetically that experiment to a clinical trial for a human. This human who is injured, receiving embryonic stem cells, now their immune system has to be compromised. A further level of complication on their injury or disease, whatever they have, okay? Because that now that puts them on an immunocompromised situation. But then the bigger issue is these tumors growing. So these embryonic stem cells, at the end of the day, typically they cause cancer because the whole embryo wants to grow there, okay? And so that's no healing. Wants to grow, okay? So the pluripotent cells, so that's why I'm saying the theoretical gold standard is too much potential. And I just make a simple little analogy. It's like having a transistor radio, when transistor radios existed a few years ago, all right, and use one of those small batteries, like a AA or a AAA battery, small battery. It's only 1.5 volts, right? 1.5 volts, the little batteries. So now I have this transistor radio, but I have a battery that is like a AA battery, but instead of 1.5 volts, this battery happens to have 200 volts in it, okay? 200 volts. I put that battery into the radio, and what's it going to do? It's going to fry the radio. It's too much voltage, all right? And that's what's happening here. They just have too much potential. And so functionally, biologically, clinically, they're not working. And that's why over the years, because this research now has been going on for decades, about between 20 and 30 years, stem cell research. And have you ever heard of a human clinical trial on embryonic stem cells? Finally, I think it was about 5 years ago, maybe older, I think Enron, was it Enron? It was one of the first biotechs that came up in the 1970s or so in California. Finally, they were able to get a protocol approved by the FDA, because this needs FDA approval to go to clinical trials, to go to human trials, okay? Bless you. And it was something kind of simple, really, certainly not life-threatening. It was, I think, macular degeneration that they were looking at to heal that with stem cells, with human embryonic stem cells, all right? And finally, after many years of tweaking the protocol, this biotech in California was able to get FDA approval. So they started the trials, wouldn't you know it, within a few months, they had to shut it down because they started getting tumors, or started getting negative results from the patients, of course, with consent and all that, but they were getting negative results. And they shut it down because they're gonna be sued out of their, out of the water for these trials. And so to this day, the one protocol that I was aware of on human trials, even that was shut down. And so it's just not happening because they just have too much potential. So whereas theoretically, yes, they are the gold standard, in practice, they're not. They're not, you just have too much potential. Better,
Why do they keep going with it? Why not focus on the
that's the twist. That's the twist because, the mind is, the beginning of Lent, I always start talking about pride and arrogance and terco, stubbornness. We'll see why. We'll see why in a minute. Better are really the multipotents.
So the church is against
Embryonic. Human embryonic stem cells. Because it involves killing the human embryo.
Would this be
It involves killing
their
the human embryo.
own 50% embryonic cells for better compatibility? Like if it's a female patient, you actually harvest their
Right.
eggs and then you get a sperm.
Imagine,
You
so
have
that's
the first
involving,
one from the-
exactly. So that would involve that patient first to be reproductively capable, then they would have to go through in vitro, right? Either with their spouse or with sperm from a catalog or egg from a catalog, etc. So it gets even more messy generating those embryonic, those embryos. So which are the most likely customers for human embryonic stem cell research? The frozen human embryos, the spare embryos, the million embryos in the United States that are in these clinics. And that's why I said, when I talked about IVF, that now that industry has found a way of making even more money with those embryos by donating them to research, to human embryonic stem cell research. And they donate in quotations, but then the industry, the IVF industry, is entitled to compensation for running all the maintenance on the embryos and so forth, which will amount to several $1,000s because that's expensive technology, high technicians, high salaries, etc., etc. So you see, but to Maggie's question, what has happened, if they don't really work for healing or regenerative medicine, they're causing tumors, right? These embryonic stem cells are causing tumors. So how about we use them for cancer research and generating cancer cell lines for research? So you see, the mind is what the mind is going to be. And for let's say, for putting a label on it, for a secular humanist who doesn't believe at all that this human being has a soul or anything Catholic or religious or spiritual or whatever, this is just an embryo, okay? So for that person, listen, cancer is the big topic today. People are dying from all kinds of cancer. How about if we find a cure for breast cancer by using one of these human embryos, okay? Or pancreatic cancer, or whatever. And so now the latest twist is to use the human embryonic stem cell research, since it's not working for healing, it's just for cancer research.
Well, what is wrong if you're going to have it? It's basically there is a better good, or at least some good that comes from something bad.
Exactly.
So if, I think the church has a thing, you commit a small sin to prevent a greater sin. So if you're saying,
Right.
if if this is going to occur,
Right.
then why not try to do it the most ethical way, the most productive way because of this supposed, which you characterize as a tragedy or an ethically wrong action?
Right.
Why not use?
Yeah.
I mean, why waste them if it is already happening?
Exactly.
Because it opens the door.
This
The
is
door's already open.
But the word,
Right.
the word.
The
is
door is
wasted.
open, yeah.
The word is wasted. Why waste?
So why do it in the first place?
But that's not the issue anymore.
The word.
right.
The issue is the barn door is open. Okay?
Sure.
And whether or not you can change it and close the door later, but now that it's actually
Right.
open, why would you
Yeah.
not avail yourself to these products?
Right.
But then why not close the door period? Not-
Yeah.
But you're
No, the point is, okay, so the point is this, the door is open and the door is wide open, but why go through that door? If that door is leading me to evil, you say a smaller sin for a greater good. The smaller sin is not a smaller sin, it's the killing of a human being.
but it's already been killed.
Right,
That's
but do I
why-
have to participate in that killing?
Well, that's a different issue.
Why, how is it a different
It
issue?
is a different issue because if you didn't participate, but somebody could benefit from that, why would you say they should not be able to benefit? I understand you have that people have this belief,
Hmm.
so they don't want to, but other people don't subscribe to it. And you don't have to participate. If you had a cancer, if you had some illness, if you had something, you could not avail yourself to the
Mhm.
therapy.
Okay, let's put a parallel example that may make it a little more graphic. There have been even some movies on this and certainly novels and so forth, but one person one day wakes up all of a sudden naked in a bathtub full of ice. And when they wake up, they read a sign. Welcome to the organ donor community, right? And then they look down and they have a surgery wound that goes from side to side. And it turns out that their kidney had just been excised. Who knows who did it? But I am on kidney failure. I'm a renal failure, stage 4, and I've been on the list for years. And I have $1 million in the bank, and I know that with only $10,000, I can buy that kidney. I know where the kidney came from. Okay? I know where the kidney came from. Should I buy the kidney? Would you buy the kidney?
Me? Probably. And I would think, and I also think that people would have the right to sell their kidney. Whether or not.
Yeah, but the person in the tub was not consulted.
That's not.
Was not consulted. Would you really buy the kidney knowing that it was stolen from another person? Would you buy it? You would. Okay. How about if that person is your sister?
Yes, I mean. if
You would
it's
still do
done,
it.
If
It's done. Knowing that the kidney was stolen. Siobhan, think about it, please. Think about it, because you're pushing it to the hilt. Okay, just for the sake of making a point that it's not defensible. If you knew that your sister, that someone stole a kidney from your sister, listen to what I'm saying. You would buy that kidney knowing that it was stolen from your sister without consent.
Yes,
You would.
absolutely.
And then you probably go and give it back to her, right?
But once, I mean, because this is such, it's a fallacy in logic to keep going to the extreme like that. But if you want to give that argument, the fact of the matter, once the kidney is out, do you waste it? Do you not give it to somebody, your sister, or take it from someone, or if you need it? Some people might say no.
You
but
waste it.
other people would.
Of course, other people would, just like other people.
I mean, that's a possibility also.
Right,
I understand what she's saying. She's trying to salvage some good
yeah,
from a bad situation.
right.
But the point is you compromise. And you're saying, what you're doing by doing that is saying, well, it already happened, so let's try to find a silver lining for this cloud. But in the meantime, there's a whole population out there watching and listening and saying, well, this happened, so I guess it's the slippery slope. So then it sort of condones it,
Exactly.
Encourages
Fine.
other people to.
It gives a
follow.
social
That's
approval.
what I did, so now we can.
Defines justification on an action that is indefensible. And this is similar to the acts of reparation, retribution. And let's say that during the Holocaust, most Jewish families, they were removed of all their goods. Not only they were killed eventually, and you cannot get that back. And even if they were made soap or something in the process, but they were removed all the gold and the jewelry, etc. And all of a sudden, it is found by somewhere, is as a property of somebody else later on that obtained it.
Yeah.
And it is found. What is the moral thing to do? To return it to the family that remains alive somewhere in the world? Or, I'm sorry, this is part of my patrimony. I am supposed to, that has value, that has material value. Should I keep it and keep profiting with it, or showing it around, or I should do the right thing and return it to the regional owner that was unlawfully removed?
Right.
I mean, these things,
Even the Holocaust
it might have
itself.
value on that person, and might use it, and I say, yeah, but I'm using it.
See, I think that the
Why
Holocaust
not?
itself is a very good example. The Holocaust is a very good example, as you mentioned it, because, you know, the intentions of Hitler really were good intentions. In other words, he was not trying to eliminate the human race. He was trying to purify the human race. He was trying to make the human race better. And anyone reasonable would say that's a good thing. He was trying to purify the human race. And what if we had to kill a few million? What if his plan would work? If by killing 6 million people or 12 million, which were actually from the Holocaust, the human race would have been purified. Wouldn't that be a good thing? You know? I mean, these people were gonna die sooner or later anyway, right? Because no one lives forever here on Earth. If they were gonna die sooner or later anyway, why? And if we knew that by killing these people, that's his error of logic is in thinking that by eliminating Jews or homosexuals or Catholics or whatever he thought was not Aryan, that would actually purify the race, okay? But let's say that hypothetically he was correct, that the weaker individuals in our humanity, and by eliminating them, we would actually purify the human race. The theory, the ideal, the goal is a good one. We want to purify the human race. You know, we'd be better people. But the means, it comes down to the means. So that's why I spoke already very clearly about the distinction between utilitarian or principled ethics. In other words, in utilitarian, a good end will justify whatever means, as long as it's legal, right? As long as it's legal. But it's hard to just phrase it in any other way than the means themselves have to be justified. And the fact that it's occurring, because I know it's occurring, so is addiction occurring and many stuff, murder, just what's today, Saturday? 2 days ago, there was a killing 1 block away from where I live, up there in Miami Gardens, 1 block away. It was in the news all over the place, all right? Now, do I need to be part of that killing? No. That killing, we can say objectively, is wrong. It's always wrong. Murder is always wrong. I heard the shots, pop, 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 pop. I was in the kitchen, and it was 1 block away at 4 o'clock in the afternoon. You know, the guy was going to die anyway, or the guy was a drug dealer. Why not take him down? Society will be better without the drug dealer. Let's take him down. The ones who killed him knew who he was, right? And people generally don't get killed for being good, right? They're doing something wrong to another gang. So let's take him down. It's going to happen anyway. You see, so the difficulty here is being engaged in an evil act, an act that is intrinsically evil.
Mess is there. Okay, it's messy. But logically speaking, for me, I don't think anybody would like to stay in a mess. When I'm looking at it, if I walk into a messy room, for me, that's like, oh my gosh, I have to clean it up. I have to do something.
Yeah.
But if I come into with that idea, oh, the mess mess is there. Let me just sit in the mess.
Yeah,
That makes
and
no
be part
sense
of it.
to me. And, yeah, and be part of the mess. Because if I don't clean it up, I might get, the mess is gonna get bigger. I might get roaches, I might get, bacteria, I might get sick because of that mess. And that ideology, that idea to come in and say, oh, the door is open. Oh, let's just try to, kind of maneuver around it. It makes no sense to me because, like, if my niece has all her toys on the floor and I'm walking and I stub my toe and I hit my toe and I'm like, oh, she's my niece, let me leave the toy there. I'm gonna keep on stubbing my toe. And if I don't want to stub my toe, I'm gonna have to eventually pick up the toys.
Mhm.
And it makes no sense to me logically for some people to just like, oh, the door is open, so let's try to
Just walk in.
walk in.
Yeah. So in other words, wouldn't it be a better deal to try to teach the niece after you play, try to pick up again after
Yeah,
yourself and do
Teach the
some
person,
teaching?
Like, okay, yeah, I took out the kidney and I'm gonna sell it on the black market, but you could have done something better with the kidney. How about you give it back to the rightful owner because the person did not condone the crime.
Consent. Did not consent.
It's like a stolen good. You're not, well, okay, we already have it. Let's keep making profit on it and continue the cycle. I found a very striking similarity. It's an analogy with justifying slavery at some point. Throughout civilization, but in our particular case in America, when some slave owners, they said, like, well, these individuals have been under my care. I'm a lawful citizen. I believe in God. I treat them humanely. But why giving them freedom now when they spend most of their life already under this servitude? And why do I have to go through the expenses of release them when I will keep protecting them by owning these people? Yeah. Why do I have to just give them the freedom? So it's a state that they already have. They have spent their entire life under that kind of mandatory servitude. And you might justify, like, well, they don't know better.
They cannot fend for themselves. It'll be better.
They were already uprooted from their origin. Why we have to disrupt the system if it's already working, contained this way? Just because it's a human.
They happen to be human beings.
Freedom. You have to do it immediately the moment you realize that maintaining the status
that they are really truly human.
Exactly, you are doing a great error, and I find that situation. Okay, the embryos are already there; they have to be maintained, but they were part of the process. And if you then all of a sudden justify use for them, you might end up, as you say, condoning what already
becomes a habit. Human nature is what it is. In fact, back then, the big argument for not giving freedom to the slave was an economic argument, that the cotton industry would collapse, the cotton economy, and a lot of the South depended on that cotton economy. And so it was putting money above people, really. Because anthropologically, no one questioned that they were human beings, but it was a legal thing. It was legally, they just didn't have the legal rights. So it's very much parallel to the fate of the human embryo today, is that they are not recognized as legal persons. So, Siobhan, I know it's challenging, but it truly, the way I see it, it's a mindset of something that we kind of grew up with, perhaps that we absorbed along the way, okay? Look, I'm with you 100% with all of the abuse that has gone on to women and to minorities and so forth. The challenge is to get into the utilitarian mindset that it's already happening, so why don't we just go with it? Because it's gonna happen anyway. Evil is happening right now, and why should we not participate with it? Because of the principle that evil is evil. Okay, but until you actually see this organism as truly and fully human as you and I don't think we're going to get anywhere. Until we actually see this microscopic organism as fully human as you and I.
Well, I don't think it's as human as you and I.
Exactly. And so that's the prior.
And it's, the potential is certainly there. It's
Yeah.
where do you assign the rights to it?
Okay.
And afterwards, like you said, even if you outlawed abortion completely today, and you still have your snowflake embryos or whatever, the snowflake babies, or
Right.
whatever, the snowflake thing.
Yep.
Proper burial. Proper burial. That's what it's.
Proper burial?
Proper burial. If they're already dead, you
Okay.
give proper. You want.
If they're dead. If they're dead, but if they're alive, they
I
remain.
don't go and get the bone of the dead and just ground it, give it to the cows because I can make money out of it.
Some people are doing that now.
And feed the poor.
Yeah.
I know the use for it, but that's not the way we go. That's not the way you want to be. You say we all get old. At some point, we're gonna be some kind of burden to society and to our families. Come on.
So,
We honor our father and our mother. We take care of them until the very last. We just don't say, you're
euthanize them. We're going to go there.
drop dead. And no, we don't do that. That's not who we are. At the beginning, in the middle, at the end, it's the whole cycle. Eventually, we go to heaven. Even after you die, there's still things happening out there, you know? So why do we have to do these actions that we know that we don't really have to? We have other means to get to that end. Why we're gonna persist in giving a use to something that it originated wrongly, unethically? It's inhuman. You just give proper burial. At the end of the story, we make mistakes all the time. I mean, there was a Holocaust, there was slavery. Imagine, I mean, we're humans. It's a bumpy road. Do we go back to the past? We cannot. Time move in only one direction.
And if we don't learn from the past,
we have to acknowledge our mistakes.
we're going to make the mistake again.
Put an end on the story.
You had a comment?
Okay, like how you mentioned with your family, we're gonna get old one day. I wouldn't want to be in a place where, okay, if I get 80 years old, hey, take me out, like just because I'm old, take my life out. If I wouldn't want to be treated like that, so I shouldn't have the right to treat someone else like that. And sometimes, we tend to forget that. Like we just said, we make mistakes. We tend to forget that. We tend to put money over people. We tend to put our own ideas over others, and not remembering that. Let me put myself in this person's shoes just for a quick moment. I wouldn't want them to take something horrible and then to redo it and to feed it to me.
That's right.
I wouldn't want others. If I don't want others to treat me like that or to put me in a position like that, so let me not put someone else in a position like that. And even if the embryos cannot speak for themselves, I wouldn't want someone going into a woman and taking her embryo, and even if she's picking it up, to just do bits to it.
Right, and you make a very valid point, because in our contemporary society, many of the elderly are forced to feel as if they are a burden, okay? And many of the elderly are now feeling like they are a burden to their children, to their grandchildren. There's no time for them, and they're getting this guilt that they are elderly, and so that predisposes them toward the euthanasia and the suicide that we'll talk about in the end of life issues. It puts them in a situation that's very precarious, and they feel guilty about being alive because they're a burden. And you literally hear it from old people. I go to homes and I go to visit old people, and first of all, they're seldom visited by their family or friends. Mostly family. There's no time for them. The grandchildren are too busy working and whatever. They don't have time, and so they feel guilty about being alive, and they feel they're a burden to society, and that feeds right into the euthanasia mentality. I mean, I'm anticipating, but we're going to look at this in the end of life issues, to the point that they rather be killed than continue to be a burden to society and to their family and so forth. Okay. And that's a very sad state of affairs. You know, the honor and the dignity of a nation and the greatness of a nation can also be measured by the way we treat the weakest ones in our society. The ones who need advocacy, the ones who need most protection. That's the true greatness of a nation, not the, what is called the GPS, the gross national product or whatever. Okay, it's the way we treat the weakest ones in our society. That is the greatness of a nation. And so here we have one of the weakest who really cannot speak for themselves. So it does come down to advocacy. It comes down to advocacy. And that's why I spend so much time at the beginning of the course on trying to emphasize the biological fact of the humanity of these creatures. So now, in a sense, this is historical, as I said at the beginning. Why? Because biologically, they're not working. Clinically, they're not working. They're causing tumors. There's an immune rejection. And so even if it's just for the legalistic aspect of not getting sued, many labs are abandoning human embryonic stem cell research for healing, for regenerative purposes. But now they're coming back around to doing cancer research with it. Okay? Now, yeah, amazing. With all the cancer we have, you know, that's the last thing that the patient needs is for the treatment to cause them cancer, right? And so people are walking away from that. Also, thanks be to God, it happens that there's a very healthy alternative, biologically and spiritually or morally, which is adult stem cell research. And that's why we make that distinction. That distinction is not without its significance, okay? It's an ethical significance because it turns out that adult stem cell research is working and is giving results, all right? And so this is the happy ending of this thing, is that some advantages at the biological level, okay? Because this is the proof is in the lab, right? The proof is in the pudding whether the cells are working or not. Is that rat getting healed or not? So they're multipotent instead of pluripotent. In other words, they're already predisposed into healing a particular organ system, all right? So that's an advantage. Less potency is better here. This is a case where less is more. This is the case where less is truly more. They're already predisposed in a particular organ system. Bingo. The next one is they can be autologous transplant. In other words, by the same patient, I have, let's say I have cartilage degeneration of the knees. Well, the doctor can take out stem cells from my circulatory system and inject them directly into my knees for the hope of cartilage regeneration. This is autologous transplant, self-transplant, right? Autologous, self. It bypasses the immune rejection, okay? Because they're fully compatible. They're 100%. I am the source of my own stem cells, okay? And they have actually been successes over the decades now because this alternative stem cell research, human adult stem cell research, has been going on for decades now. And there is one, at least one outfit that is devoted to this. It's a group of clinicians and ethicists and so forth. Their name is Do No Harm. They formed a team called Do No Harm. It's housed in Virginia near Washington, D.C. But if you just Google this, org, I think it is, it's in .com. This is stem cell research. I have a website. The team is called Do No Harm, which is the first principle of medicine, right? And they have this website where they post all the journals, all the scientific articles that have come out over the decades on successes with adult stem cells and the zero successes with the embryonic. See here, Right now, they're up to 73 successes in adult stem cells, say we're on the embryonic. You can go here, and these are the actual articles. Now, it's at different levels of trial, all right? Different levels. You can explore the site in more detail, but at least there's one site that is tracking this, that's tracking the successes, and this is at the clinical level. Different levels of success. Many of these are still in clinical trials, but the interesting thing is that it's working. Peer review articles. All right, these are all the citations. Back to the late 90s, it's been going on. All right, so at least we have something positive going on, which is a healthy alternative, and a case where the gold standard hypothetically has been proven not to work in practice, but the alternative is stem cells from our own body, because we have them. We have them, for example, in the pulp of baby teeth, right? That's why we get new teeth. We have them in the follicle of hair. We have stem cells, neuronal, which is a challenging one because typically neuronal cells last for years, and when they become fully differentiated, it's hard to find stem cells, but they have found stem cells on the hippocampus, which is in the center of the brain. It's a gland there. Apparently, oxygen stimulates that hippocampus to produce new neuronal cell lines, okay, as cell types. Now they found, well, bone marrow, which we didn't know back then, but bone marrow transplant is actually stem cell transplant for hematopoietic cells, in other words, cells in the circulatory system. And now even found stem cells in circulation. Now we have stem cells in our circulatory system. So a process similar to phoresis, I don't know if you've ever done phoresis, that's to obtain platelets from the blood, okay? It takes several hours. I've done phoresis a couple times. I used to be CMV negative, so that qualifies for giving blood to babies, all right? And then at some point I became CMV, it's a cytomegalovirus. We start out negative, and then at some point we're exposed to the virus, we don't even know it, we have no symptoms, but we become CMV positive. And so that loses our ability to donate blood to babies because they're still sensitive to the virus. At any rate, when I was CMV negative, I called for platelets for babies. And so I've done phoresis a couple of times, and it goes on for hours, about 3 hours more or less. And you talk about getting cold because they put the blood through the machine. And it's something similar to dialysis, functionally similar, that the blood is going through a machine, and the machine is filtering, literally filtering out the platelets live. Okay, so they're spinning the blood. Imagine the sophistication of that machine that is filtering the blood for its platelets and then pumping the rest of the blood back to me. But they have to do that process in the cold. So they're pumping blood back to me cold, literally cold-blooded. So I keep getting colder and colder, and they put more blankets. By the time I'm done with the process, about 5 or 6 blankets on top of me. Because one keeps getting, yeah, exactly, just like here, right?
What was the organization, do you know how.org?
Right, no, stemcellresearch.org. I have the link here. Stem cell research, single word, stemcellresearch.org, okay? So they're tracking it, and so that's a positive thing. So now we have something positive, a healthy alternative. Of course, all this implies that what's the one thing that still needs to be in place for adult stem cell research, human adult stem cell research? Consent, all right? So they should not take out, it's so obvious that we take it for granted, but we should not take out stem cells from people without their consent, okay? And this would be unthinkable here in the States because we're already of the born, but we can think of a number of countries where this could go on without their consent, right? Analogous to taking out organs without people's consent. So as long as consent is in place, we can go forward. And so it's very, very successful, right? And okay, so enough said about adult stem cell research, a good alternative. Yeah, please, let's take a break. All right, so let me talk about, in the meantime, Yamanaka, I was doing my, okay, let me do this because it is 11:30 almost. We're going on for a couple hours. Let's take a break and I'll pick up here as soon as we come back, okay? Because for human cloning, it's gonna be faster, shorter. So why don't we take a break now, about 10 minutes? And then return to finish up the stem cell research. We'll talk about IPS and we'll talk about cord blood stem cells. All right, so let me pause and we'll be back in about 10 minutes. Okay, hello and welcome back. Welcome back. We'll go for the second half. I forgot to mention at the beginning of the course that this is on the beginning of human life. We're covering today, obviously, stem cell research and cloning and human cloning. So moving forward, I still have two other types of stem cell research to cover, which is the induced pluripotent stem cells, and then finally the umbilical cord cells, umbilical cord. So I don't know if any of you had heard of Yamanaka, Shinri Yamanaka, but he was a scientist in Japan that was doing human embryonic stem cell research back in the 90s and into the 2000s. And I was doing my doctoral work in genetics at Purdue at the time. So this was all over the news. It turns out that he was looking at these human embryos when he was disaggregating them under the microscope in the lab to get the ICM to onto the Petri dish for generating those cell lines, right? He thought to himself, because he was interviewed afterwards by major news services of the world, he thought to himself, these blastocysts that I'm disaggregating look very similar to the way my daughters looked back when they were human embryos, kind of paraphrasing what his thought process was, you know? So when I was reading this, I said, wow, this is an honest person. He was looking at human blastocysts under the microscope, disaggregating them to get to the ICM, and he's thinking to himself, these blastocysts look like my daughters looked when they were blastocysts. Okay, so he's recognizing the humanity as that. And then he said, there has to be a different way. There has to be a better way. And so he started pursuing this other way, which was to isolate the factors, the proteins that are developmental proteins that regulate embryonic development, to see if he could undifferentiate or de-differentiate differentiated cells, okay? So there's a lot of differentiation here and de-differentiation. So it's becoming like a tongue twister. But the idea was essentially, this is what came to his mind. What if this is a natural process, is to go from pluripotency eventually to fully differentiated cells. It's like a one-way street, right? One-way path in nature from undifferentiated cells to fully differentiated cells to function in their organs and tissues specifically. What if we can reverse this process? In other words, what if I find the factors typically meaning proteins that are regulatory proteins that are guiding, that are regulating this process, this pathway? And what if we try to reverse this process? so that we could take cells that are either fully differentiated or multipotent, because at that time the gold standard was the ICM, and reverse them back to their pluripotent state, but not to pluripotent. In other words, not reverse them back to human embryos, but just stop at the pluripotent stage. In other words, what if we're able to take fully differentiated cells or in multipotent cells and back them out back to their pluripotent state, and then redifferentiate those cells into a different line? So Frank, take cells like muscle precursors, undifferentiate them back to pluripotency, and then redifferentiate them, say, into neuronal cells. Okay, so we would go from one system to another. He wanted to change tracks, essentially, go from one organ system to another. Let's say from circulatory system to nervous system, which is really the big challenge is in the nervous system. Okay, so that was the idea. Go backwards against nature with factors, regulatory factors, the regulatory proteins that normally guide this process. That was the goal. Well, other labs of the world got onto it also, and there was a race going on. And he was spending 16 hours, 18 hours in the lab a day with his whole team until he finally got it. And he got it. He was able to identify the factors, which I think come down to about 3 or 4 factors, 3 or 4 proteins, regulatory proteins, that regulate the differentiation process, And he was able to get fibroblasts, for example, which are in the mesoderm, to undifferentiate back into pluripotency and then get them into another cell line. So he was jumping from one cell line to another by undifferentiating cells and then redifferentiating them. That's why this method is called induced pluripotency, because that's exactly what he's doing. He's inducing cells to become pluripotent, from multipotent or fully differentiated to become pluripotent. But it's done artificially in the lab, okay? And that's why they are being induced to do that. Again, against nature, against the flow of nature, all right? And so they're known as iPS cells for short, iPS cells, all right, or induced pluripotent stem cells. So for convention, the I is put in, lowercase, because it refers to the adjective, what is being done to the cells themselves. So here's the man. And what motivated him, again, is essentially recognizing the humanity of this blastocyst. They look very similar to the way my daughters looked when they were that age. Okay, and this is a man who, I don't even know if it's Shinto or not, but certainly not a Christian. He's doing it on the science, on the biology of it, on the biology. Okay, so that's iPS. Then there were some issues with it at the technical level, because think about it, physically, physiologically, what does this involve? He has to, so first was the isolation of these factors, which I think came down to about 3 or 4 factors or proteins, regulatory proteins, right? He had to isolate the proteins, but then he had to inject those proteins into cells, into either multipotent or fully differentiated cells, like the blastocysts, okay? The fibroblasts. How do you do that in the lab physiologically? What is used as a syringe typically are viral capsules. Viral capsules. In other words, we insert the proteins inside the virus. First, we excise the virus of its DNA or RNA if it's retrovirus. I don't know what they use, but they use essentially the viral capsule as a vector, as a deliverer, as a syringe for putting those proteins into cells, all right? Challenge, because viruses, you're doing this on a statistical level. We're not actually engineering each individual virus. We have a vial with trillions, quadrillions of viruses, and we are processing them so that they take in that protein, all right? Let's say we do that to a 99.99% purity, well, that still leaves a few viruses intact with the original DNA or RNA, which is an infection. And so, being a statistical process, there was always the danger that a few of these cells would become infected with the actual virus RNA or DNA, okay, instead of the protein of choice. And so that was a technical issue there that had to be resolved. I think eventually they were able to work around that and have capsules that were pretty much 100% pure for delivery. Mm-hmm. There was also the danger that some of the factors of the virus would also get into the cell, and that had to be dealt with and so forth. So it's been purified since. So now there is a possibility of obtaining cell lines from multipotent cells by using this technique of IPS. All right. So it was a big success, at least at the time, and it was all over in the news. I think he might have even won a Nobel Prize on it in medicine or something. But it's another viable, healthy, and ethical way of doing this, trying to achieve that gold standard of the pluripotency, at least to change tracks into another cell line, from one cell line to another. Another one that is, again, natural, all right, and does not involve something unnatural as this, going backwards on cellular differentiation, is the cord blood that I talked about briefly a little bit, okay? And so let's look at that for a moment. When the baby is born, there is typically there's the placenta, the umbilical cord, and the baby, and that all forms one complex unity that has to be severed, right? Because I always like to comment that the placenta, first, does the placenta belong to the mother or to the baby? I know there's an interface and there's maternal contribution to the placenta and so forth, but essentially the placenta belongs to the baby. In other words, the mother is not directly or vitally dependent on that placenta. She may benefit from it and so forth, and there's back and forth. We know even cells, embryonic cells end up in the, or fetal cells end up in the mother's body, and maternal cells end up in the fetus, okay? So even cells can get through, but also chemicals, the immune system, et cetera, et cetera. But at the end of the day, the placenta really belongs to the baby, and it is a vital organ, but temporary. It's a vital organ that is discarded at birth because it's done its function. So it is a vital organ, but it's only during the embryonic stages. And then it's discarded, becomes medical waste at birth. The cord typically also goes the way of the placenta. So in other words, the cord is cut, and the knot is tied, put inside the belly with a tape, and that becomes a belly button. So the umbilical cord typically is thrown out as medical waste, okay, incinerated, etc. But it turns out there's blood inside that is mostly baby blood. And that blood also has stem cells in it. And so the interesting quality about these stem cells in the cord blood, even though it's a little blood and they're few, but they're real and they're pristine in the sense that they have embryonic quality. You know, they're tender, they're fresh, they haven't been exposed to a lot of UV or anything else like adult cells, all right? So they're more pristine, all right? But they belong to the baby, so they're 100% compatible with that baby, and a good compatibility also with siblings of that baby, because they come, assuming that the siblings come from the same parents, and also at least 50% compatibility with the mother and the father because of the DNA. And so the immune issue is toned down because of that percentage compatibility, all right? So there's quality there, there's biological value. And those stem cells can be coaxed into becoming cell lines for different types of tissues. They're called mesenchymal, all right? Mesenchymal stem cells. And these are precursors of cell lines of various tissues of the body. Okay, so bottom line, the stem cells in the umbilical cord can be used later. So the hope is that they never be used. In other words, that that baby never has an accident or an injury in his or her life. But if they do, they have these stem cells from the umbilical cord that were saved when the baby was born. Okay? So the idea is to save is not the cord blood, is the stem cells inside the blood. But typically they talk about cord blood or cord cells, referring to the umbilical cord. All right? So they are umbilical cord blood stem cells. And in the normal literature and parlance, you see basically cord cells or, yeah, cord stem cells, referring to the umbilical cord blood. There are the cells inside the umbilical cord, and they're ethical as long as we obtain the consent. The baby is impossible to give consent, the parents of the baby. And why is it reasonable to obtain consent? Is it hurting the baby? Not at all. That thing is going to go to the incinerator. Okay? So the baby is already born, and that's the crucial issue there. The baby is born. There's no harm at all to the baby.
It's been a long time since has been identified as you know, those potential uses.
Yep.
Farther back or later.
Mm-hmm.
However, how is that there hasn't been any political movement towards trying to get insurance companies
Exactly.
help you with the burden
Of the payment.
preserving? When I have my children, I knew it was offered.
Yeah.
I just couldn't afford it.
Exactly.
Just couldn't.
So we have a real life situation of,
It hurt my heart
right.
because now my son has a condition,
Really?
an autoimmune condition
Oh.
in his eyes,
Ah.
uveitis.
Uh-huh.
Usually, it evolved into juvenile. It's kind of arthritis. How do you call it?
An arthritis type?
It's rheumatoid.
Oh,
arthritis,
rheumatoid
but
arthritis.
It's
Yeah.
not a typical rheumatoid.
I see.
Idiopathic or juvenile because they don't know the causes, but it's an autoimmune disease.
It's an
Usually,
autoimmune.
It starts as a uveitis, it generates into macular degeneration,
Wow.
glaucoma.
So he could lose his sight.
Oh,
at some point.
absolutely. If you don't
Wow.
catch it, and we were so blessed
Uh-huh.
that there's a requirement in elementary schools
Yeah.
to go through the ophthalmologist.
Yeah.
And
Nice.
that-
Very nice.
that-
Look at that.
it
That was a
he
trigger.
wasn't a specialist, but he documented something. He noticed
Right.
the changes, the cloudiness, and
Mm-hmm.
essentially the autoimmune situation
Starts-
site that was happening there,
Mhm.
and gave us a referral to the Boston Palmer.
Yeah.
We went there and we were able to stop it. We
Boom.
have some damage.
Yeah.
It's irreversible.
I see.
But we were able to put him on medication, and he's involved in fine his control. We don't know
Yeah.
what's going to happen later. For
Right,
the rest of
right.
his life, we just have to have that. But I was thinking at that time,
Mm-hmm.
I should have done it because you never know.
Right.
No,
That could have been because at the end of the day, it's a genetic thing that's going on.
in the
Right?
future,
Mhm.
but it was so expensive.
Right.
And it was just in covered
Yeah.
by my insurance.
Exactly. So this is what I've heard, that the kit, you buy the kit online and it's maybe $1,000 or so, big markup. And then you tell the nurse and the staff and even the OBGYN, the partero, whatever, that you want to save the cord blood, give them the kit. They will do the procedure, which essentially is just extracting the blood and sending it to the lab for spinning and differentiating or isolating the cells, right? And then there's a storage. Yes, I think they're basically cryopreserved. They're going into liquid nitrogen and preserved indefinitely. And then there's a monthly maintenance fee. Could be $50, could be $100, who knows? But that's adding up year after year after year And then as the child grows into adulthood, now we're talking decades of paying that maintenance, all right, with all the markup that occurs into that market because it's so specialized with highly qualified technicians and high salaries, so it's a big markup. My quandary is this: if this could lead, and none of that is covered by insurance, okay, because it's kind of preventive medicine, but it's because it's considered experimental, maybe it's not covered. The point is this: why, if insurance could benefit from this, what would it need to, in a market economy, what do we need to show to the insurance company? The dollar sign that they'll make a profit on this, okay? The bottom line, to me, the bottom line is this: if this saving the cord blood is going to lead to healing, then that's less insurance. That's that the insurance has to dish out for that child insured. They would save insurance money on the healing process because this is better healing and deeper, just better healing for the patient. So I'm thinking dollar-wise for the insurance company, it behooves them to get into funding this kind of thing because it would save them money in the long run, no? In their patient population, in their family population. But still, to this day, I don't know of an insurance that does cover the saving of the cord blood. And it does become, and when you think of typically younger families, they're having their first child, second child, they're still in their 20s or 30s, they just can't afford the maintenance first, the startup or the upfront, and then the continuing maintenance of the cells.
I think it's like something a year, so it's not an expensive.
For a year. Uh-huh.
I don't know how much you pay, for the
Initially,
for the-
yeah.
I don't think it was more than $300.
Really? Okay, so it's gone down. I have old prices. Okay.
10 years ago was
It was, huh?
10 years ago,
10.
and there wasn't a mechanism. This is an event that it happens long. You
Right.
caught it.
Yeah,
Yeah, they do so fast.
So
It
it's not
is,
only
yeah.
there's no good information structure
right.
to do it, should be recommended by the hospital. Just because of
Exactly.
that potential, but it was more expensive at the time. Or,
Yeah.
I mean, right now,
Mhm.
we have time.
Right. In hindsight,
Imagine
imagine if it has the potential to save the sight of a person.
That dollar amount
Mm.
definitely adds up because we have to go like every 3 months
Mm.
for the checked out, and there's
Oh, yeah.
specific tests that they cost a lot of money that we have to
Wow.
put up
Okay.
some
Right.
payment.
Public cord blood banking is completely free, but private cord blood banking costs between $1,400 and $2,300 for collecting, testing, registering, plus between $95 and $125 per year for storing.
Wow. Mm-hmm. There you go. You see? So yeah, it is. It's a market economy thing, So there's a demand. There's going to be a supply.
I would do it.
And- You see?
Well, what about
Yeah.
people who have a second child for the purpose
Yeah.
of curing a genetic disease, because they want, is that ethical?
There's now you bring it up. I remember a case years ago when IVF was just getting started. As I think it was actually the grandmother who was the surrogate mother for her daughter's fertilized eggs for a second child because the first child, the only child at the time, had some kind of genetic defect, and they were looking for a match because they had not saved the cord blood and they wanted to save the cord blood. So the woman had a second child on purpose just to extract the cord blood from the siblings. And that second child was manufactured in vitro specifically just to serve as a donor for his sibling. Well, the process, the in vitro process was evil, plus the grandmother becoming the gestational mother of her grandchild, for that child. In other words, that second child was manipulated to serve as a donor for his, let's assume it was both boys or both girls, I don't care, for her sister, and then when that child is born, that second child is born, he's got two mothers, and one of the two mothers is also his granddaughter, his grandmother or her grandmother, and where's the fair and where's the justice on that? You see, there's no concern for that second child. That's the utilitarian challenge, is that that second child was being used for the benefit of the first child. The intention, the goal was absolutely stellar, excellent. Want to heal the first child for sure. But in the meantime, that second child is...
The first child should suffer and die from the genetic disease.
First of all, who knows that he's suffering? Who knows that he's dying? Okay. In fact, I'm going to hypothetically make her a woman. So who knows if she's suffering, first of all? But let's say she's suffering. We're all suffering, okay? Who knows if she's going to die from that disease? She may become blind but not die from the disease, all right? Or if she dies from the disease, the point here is to use another human being for the sake of saving all of humanity. Well, that was Pilate's argument for Jesus. Better that one man die for the sake of saving the whole of the people of Israel, even if the man is innocent.
We didn't have a problem with that. That's why we have Jesus Christ.
Yes, exactly.
He
So,
was used to save his people.
yeah, thanks be to God that Pilate killed Jesus so that we could have redemption, right? he was going to die anyway. So.
He was like created specifically for that one mission.
Right. You
So
see?
it's not like, oh, we're gonna kill him because.
Yeah, but they didn't know that.
Yeah, they didn't know that. They didn't
Playing
believe
it out
that.
didn't really know that.
They didn't believe
Of
that.
course. And there was also the possibility in God's plan that everyone would have converted to the message of Christ, and then therefore he would have died of old age instead of
Going back to the argument,
being killed.
You're assuming there is no pain or there is no suffering.
But even if there is,
But if there is,
Okay,
you
if there
can
is.
alleviate that.
yes, okay, but we can also alleviate, you're a nurse, we can also alleviate pain and suffering by analgesics, right?
Well, I don't know, you can use pain and suffering. Yes, we can, but a genetic defect that
Yeah.
causes some terrible problem or so. If
The worst that we can think of, ALS, where the woman is trapped in her own body. She's going to die fully conscious of being unable to breathe. How gruesome can you get? How terrifying. So just by killing this one embryo, this ICM, this human ICM guarantees me the possibility of curing ALS for all of humanity. Should I do it? It's very tempting. And it's a frozen embryo anyway, so she's going to get flushed down the toilet someday because her parents abandoned her. You see, load the bases, right? In baseball, you load the bases, you're in the ninth inning, you have a full house, and you're the fourth batter. That's why you got principled ethics over here and you got utilitarian ethics over here. The principle has to be justified. The means have to be justified. And we're stuck. We're stuck forever watching the suffering of that person. And accompanying.
God has given us the capacity to alleviate that suffering, and so that's why
Yes.
there might be utilitarian ethics. That God has given us the capacity to be kind, to care about another person, to
Okay.
alleviate that suffering. And so if he didn't want us to do it, maybe we wouldn't have the capacity, or there would be absolute arguments
Right.
and
Okay.
prohibitions on utilitarianism.
Okay. Do you realistically think that we could possibly alleviate all human suffering?
Not all.
No. Okay. It's just not realistic, right? Even if we stack it up to the hilt. So if there's going to be some human suffering, do you believe there's such a thing as redemptive suffering? Is there any meaning to suffering? There is. So then, why should we deny redemption? Why should we deny the possibility of redemptive suffering?
Well, how much suffering is redemptive and how much is just
Yeah, on a
acceptable?
scale of 1 to 10, right? Who knows? And
Maybe I'll
suffering
take
is so,
6, but
yeah.
at 10, it's just not, it's,
well, who knows? Who knows? Heroism is part of human nature and is part of the best of human nature. So what I'm trying to say is that human suffering is so subjective, right? One person suffers watching a scene, another person can be unfazed by it. When I see someone else in pain, it causes me suffering. I'm not having the pain. My wife is having the child. I'm not having the labors, but I'm suffering watching her in pain, right? Now, some man may be unfazed with that. Another man will feel so bad about his wife that he may faint just from watching her in labor. It's so subjective. But my point is that if there is meaning to redemptive suffering, then that's the ticket. We have an alternative. We have an alternative to offer to this person in accompaniment. That's what Pope Francis is talking about, the ministry of accompaniment. If I cannot alleviate your suffering, at least I can walk with you in empathy and suffer with you. That's the redemptive suffering. And it's at the root of Christianity because Jesus is the one human who didn't need to suffer anything. What guilt did he have, right? And so in his pain, he's accompanying us. So just perhaps reflect a little bit on this. And again, is it coincidence that we're entering the Lenten season and we're talking about redemptive suffering, right? How rich it is that we can consider that there's value to redemptive suffering. And whereas we never wish suffering or pain on anyone, right? When it does come invariably, because life happens, we can accompany that person in their suffering, all right? And then there's redemption with that.
Well, you may say, I don't doubt that people suffer with other people, their loved ones, people, what have you.
Yeah.
But I think truly until you actually suffer the actual pain, it's very easy for me as a nurse to say I empathize with somebody who's dying.
Right.
Until I'm actually dying,
Okay.
it's very easy for me to say to other people,
Absolutely.
you know, your loved one is gonna, you know,
Right.
is critically ill and dying, and
Yeah.
this is for the best, or this is God's will. But until you're actually in that space,
Absolutely.
then it seems, I don't want to say dishonest, disingenuous might be a better word for it.
It could be, yes. Right,
To kind of assume that because I can accompany somebody's suffering,
right.
that it's okay that they suffer. And if there were a way to alleviate the suffering, that might be better than both of us
Sure.
suffering.
Absolutely.
Me doing the accompanying and you doing the suffering.
All right. Again, you're a little into the theory and the practice, right?
That would be against, I disagree a little bit on that argument. I think we have a lot of potential capacity to comprehend and get into the other side to experience the situation that people are going through. Because I think that we humans, we have that intellectual, mental, emotional capacity. Maybe a chimp cannot do it. That's what makes us different from the rest of the animal world. They cannot project themselves in a different set of circumstances. But we have the capacity. Just think when you're reading a book, and just by reading words, letters in that book, you are actually able to be transported into the reality of that book. It might be even
That
fiction.
novel
it might
description.
be even fiction, and you might even know it.
Yeah.
But you're reading the Lord of the Rings, and you are in the word. You're able to put yourself.
The capacity of human imagination.
Understand the suffering, and you know, the happiness or whatever of what is being in that story. So why not you as a nurse are gonna be able to see that suffering, that visceral issue that person is going through? You have the capacity. And you have the capacity precisely because all of us would go at some degree through that process. It's a natural process.
Yeah.
The process
Everything
of
has been
dying.
equal, you know, we
Right.
die of old age.
So I think that is a little bit like, I don't know how to put it, but I think that we do have the
I
capacity
didn't
to
say
see
we
it.
don't have the capacity for this. I said, we have the capacity and you can do the accompanying suffering. But I don't think people actually experience, or most people, or some,
Right.
the actual suffering that a person endures when
Correct.
they know their own mortality, or they know their own pain,
Yeah.
or whatever. I mean, I'm not saying we can't be empathetic, sympathetic.
No,
We
I
can't
think
try,
that we
but-
do.
Right, it's so personal, an individual.
That,
Yeah.
and if you could alleviate that suffering for a person, I mean,
Right.
to put possibility listening to your son's
Illness,
autoimmune
yeah.
disease. What wouldn't you do to?
Everything but hijack something of similar value. So, for example, let's say, because it all goes to where you put value in. Let's say that it's not that inner cell mass, is a little bit farther, just by chance, just because we don't have yet the technology to get to that tiny detail. And we find it at the moment of, I don't know, that the embryo is like 4 inches big.
The second or third trimester.
At that point, would you be willing to still sacrifice that creature to benefit from it? Understandable. The point is not to destroy it. I mean, you're not setting, a course just to destroy that. The goal is to benefit from it. To save another life that you put real value and emotional connection to it, like a family member. Would you still do it? Or maybe not like this, at the moment that they are 9 months before just being born. Or at the moment that they are with a little child that you need to use. At that moment where you place value.
That's where you place the value. But if somebody else, what another parent is suffering because of their child, because you've experienced it, you can know far greater than I could. I don't have children or what someone is. But if somebody said, I could not do that for my child, I don't have that value, I will do anything to alleviate the suffering in my child, do you have the right to tell them no?
I have to be part of it.
I would put it this way, Siobhan, not the right. I wouldn't have the right, I would have the responsibility to try to tell them no in a respectful way. I'm not going to incarcerate them, I'm not going to tie them down to a chair, but I would see a serious responsibility to try to get that person to see the limit of doing everything, right? The limit of doing everything. I think I would have a response. That's why I'm teaching, because I've accumulated all this knowledge of bioethics over the decades with the studies here and there and so forth. So now I feel the responsibility to try to communicate some of this knowledge in an intelligible way. Otherwise, I could just stay as a priest in the parish and keep my knowledge and keep reading and enjoying my knowledge and keep it to myself. Why share it, right? But now that I have this knowledge, now that I see the human embryo as human, I need to share that. There's something that moves me to try to share that with others who still don't see it for whatever reason. Okay? But I want to go back to that earlier issue of empathy that you put. Unless we actually experience that human suffering, it is truly very difficult to fully identify with it. I would say more that even in the medical profession and nursing and dentistry, in that whole clinical education that you all receive, I understand that there are even courses that teach the clinical professionals and paramedical to, on the one hand, empathize with the patient, but also it's a balance here between keeping a professional standard so that you don't emphasize so much that you actually become the patient, all right? In other words, at some point, the surgeon, when the surgeon cuts up the belly, he or she has to be professional about it and not feel so bad about opening someone up that they just won't do the opening, okay? And so I'm saying that there's that balance, and how do medical professionals maintain that balance? On the one hand, being an excellent nurse professionally, but at the same time empathizing with this patient who is dying of terminal cancer, okay? And I still have to administer the medication, whatever, but it's the patient who's dying, I'm not dying. And so how to maintain that balance between the professional standard and at the same time really, really empathizing with the patient and their suffering. I don't think it has a straight up answer. I think each individual has to make that, but I have seen in my years of visiting nursing homes and visiting hospitals and everything else, that some of those medical professionals and paramedical become hardened to the reality of the suffering of the patient. To the extreme that in some homes, for example, you see the patient leaning over on the hallway, you see the hallway full, lined up with wheelchairs on one side of the wall, on the other side of the wall, and all the patients are leaning over, for hours. And the nurse is over here, or the assistant is on the computer, on the cell phone all day long. And these people are just there, just waiting for their death. And so that's the other extreme. That wall has become so hardened that I don't see the suffering in front of me anymore. And it's kind of a survival mechanism because these people are dying every day. And if I empathize too much with it, it's too draining. It's too emotional draining to go home every night having lost another one of these dear ones, right? And why are their own family taking care of it? Why am I, who am a stranger to all these old folks, have to do the job of cleaning their behind and everything else when their own family is not doing it? So these are the human factors, right? These are the human factors of how we engage in suffering of others. But certainly, at least from the principal perspective, one option, which is not an option, is to load up the syringe with the overdose of the potassium chloride, boom, 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 and just give them the heart attack because they're suffering anyway. So we put them out of their misery, or they're putting them out of my misery.
That's the real question, because it becomes, which is the true suffering? You see the person is dying, or they're gonna die already, so let me just speed up the process.
Exactly.
But then, like, who is really suffering? Then it becomes two families are suffering. The person who died, and then you get caught, you're gonna go to jail. And then your family begins to suffer as well. So it's like a whole cycle of suffering. Why not let it happen naturally and let the person pass away naturally, but then you're not gonna also, come into that whole cycle of suffering.
Become a participate, become a collaborator in the agency. You see, that's a challenge. We'll see that. We'll see the distinction, crucial bioethical distinction between killing or allowing to die. Killing or allowing to die. It's a crucial bioethical difference, whereas one is not acceptable and the other one definitely is, because that's what we accompany the person. So we're always back to this core issue of justifying the means in themselves, all right? And that's what the rub is. Some people just, they don't see it, they never had an education, or who knows, their circumstances, they were brought up in a particular way, etc. But the means, it's always about the means for us. So, having said that, let me move into human cloning for a moment. Cloning, okay. So with stem cell research, the bottom line here is that thanks be to God, there are options that are healthy and ethical and are successful. And it's a work in progress. I would also encourage parents to send letters to their insurance companies to try to get them to at least start with a percentage.
Why don't
of the
Catholic
cost.
hospitals do it as a matter of course?
I would think, yeah.
But they
I think,
don't.
exactly.
So,
And why don't they? Perhaps it's ignorance, who knows? I really don't know. Maybe I should approach. We used to have a Catholic hospital in town, now we lost it. But, what do you got? Human cloning?
Is that the word, technical word, or reproductive?
Pro. Yeah, sorry. Thank you. Yeah. Thank you.
some of the largest hospital systems in the country are Catholic.
Yeah, it amounts to about 10% of healthcare in the nation is Catholic healthcare. So yeah, now who absorbs the cost? Because the cost is still real, and you're saying basically we should just absorb the cost for the principle of it. Yeah, it's nice and it sounds real good, but at the end of the day, who's going to pay that? well, maybe the parishioners are paying with their contributions that goes to the ABCD or whatever. Maybe a fraction of the ABCD campaign of the Archbishop should go toward sponsoring that, at least for, again, thinking aloud, the young families who can't afford it. That would be a nice gesture, okay? So that could possibly go toward a good PR and a good cause. But, and perhaps poor families or families with fewer means, let's say, may have some access to that. Maybe, I don't know, but maybe, again, thinking out loud through Respect Life office or something, be something worthwhile investigating in the Archdiocese if there's any provision from the Archdiocese to help out these families that are trying to save their corporate. Yeah. It's a nice little research project. If someone wants to look into it, and may count as part of the internship.
Really?
Yeah, absolutely. In fact, I'll give you a lead. Catholic Health Services. You can be an advocate there. Catholic Health Services is a big organization. They have a large budget. Catholic Health Services. And the head of that is Joe Catania. I read it for you. I'm part of the board of Catholic Health Services in the Archdiocese of Miami. So that's a good little start there. And with your nursing background, I think that this could be something very exciting. Of course, it's going to get Father Choff in trouble, which I just love. So
I don't know he's gonna like me.
go for it. And he's the head man. He knows me well. We've known each other for decades. You can give him the reference. He even knows about this program because I try to advertise through them. But this is essentially the umbrella organization of the Archdiocese of Miami that covers our nursing homes, Villa Maria.
Can I reach into the chancellery?
Yeah.
Okay.
There may even be a website.
I mean,
Yeah.
because I don't find their answer in their system.
Yeah, don't try the phone. Nobody answers phones. It's horrible. But get into the website, and there should be some stuff out there as a beginning. Okay, we run nursing homes. There is St. Anne. There are hospitals. There are rehabilitation centers. St. Catherine West, for example, down in Hialeah. So there's about a dozen places. Dominic Gardens, for example, is a building for elderly.
They're in the Catholic hospitals here.
Not as such. We used to have Mercy Hospital. Now it was bought out, but they're maintaining the Catholic identity. So that's it. Sorry? Holy Cross, but let's see. Holy Cross.
Been taken over by a different entity, I believe.
Yes.
No longer, it was the Sisters of St. Joseph.
Exactly. Sisters of St. Joseph, and they had to let it go. No vocations. And so that's gone. Yeah. And now, but this system, which is CHA, I think it's called, it's a huge system. I just want to see, HCA, thank you, HCA. HCA is one of the largest healthcare systems in the United States. I think they have about 120, 140 hospitals. See? Yeah, exactly. And they own now Mercy Hospital, and they've maintained the Catholic identity of Mercy. They also own Plantation. So this is a national. So St. Mary's, okay.
No, St. Mary's could stand.
Right, but my question is that overall as a company,
Yes.
they have a Catholic identity or just the
No,
institutions
this one in particular, Mercy Hospital, it was part of the initial agreement that they will hold the Catholic identity for 5 years, basically maintaining the ERDs, all right, for 5 years. And if it was still profitable, they would continue afterwards. And the 5 years were over. I think they're into 8 or 10 years now with the buyout. But again, in dollars and cents, what does it mean? That they cannot do therapeutic they cannot do sterilizations, no contraceptions, no abortions, of course, so no in vitro, especially because that's a very lucrative industry. That's what brought down the possible merger between Mercy and Baptist when they were both standalones. Okay, Mercy was just a Catholic hospital there, Biscayne Bay, and Baptist, the original Baptist Main on Kendall Drive, was also standalone. And back in the 90s, there was talk of a merger between the two. And that merger collapsed precisely because Baptist would not let go of an in vitro clinic that they had that is very successful, okay, very profitable. And so that eventually Baptist then went on to become a system of itself, and now it owns about 8 or 10 hospitals in the county, and it's gone into Broward now, I think, Baptist and Palm Beach is expanding. Yeah, it's the whole thing. You know, the market economy, you either expand or you get eaten out, you get bought out. And so it's locked in to continue to grow. In the meantime, yeah, it is, it's huge.
I think Rick Scott used to be.
Yeah,
Yes.
the largest Medicare fraud in the country was done under Rick Scott. He was
Oh, yeah?
HCA.
And he was what?
He was the
CEO
CEO.
for this?
For HCA.
For HCA.
Yeah, he was the chief executive.
For
Ah.
HCA,
For HCA. Really?
I remember that he wasn't that.
Oh, when he was running for
$300
the,
million worth of Medicare fraud.
oh, really? Okay, under him.
Yes.
Wow. Okay, another controversial figure, another controversial politician.
Over 500 times when he was investigated.
He did what?
He took the Fifth Amendment over 500.
500.
Yeah.
There's a tradition of that in American law, right? Yeah, wow. Okay, anyway, this is a system that took over, and at least on Mercy, they have maintained the Catholic identity, you know, even beyond the stipulated 5-year contract time. So, okay, back to this. I don't even know why I was mentioning all that when we got into it. Oh yeah, the Catholic Health Services of the Archdiocese, and that nationally Catholic Health represents about 10% of the health care in the United States. Human cloning. All right, so human cloning can be done for 2 purposes, either reproductive cloning or therapeutic cloning. The adjectives here are kind of theoretical in the sense that none of these has happened yet to date, even though cloning itself has been around since the 90s. Mammalian cloning I'm talking about with Dolly the sheep. And what do we have? So the purpose, the end, the objective of reproductive cloning is to have a baby, to have a cloned baby, all right? For a couple to have a cloned baby. The purpose of reproductive cloning is not to have a baby, but to get the human embryonic stem cells. In other words, to do cloning so as to have a blastocyst with a particular DNA, particular genome, and then use that ICM for therapy. So that's the reproductive cloning. You see that the adjective then is qualifying the purpose, all right, the end, but we still have to look at the means. So look at how cloning is done, cloning itself. Another name for cloning is somatic cell nuclear transfer. That phrase is actually telling us what the event is, SCNT. You see it in the literature, SCNT, or sometimes just NT, nuclear transfer. Okay, but basically is taking a somatic cell. In other words, it's not a gamete, and the nucleus is going to be transferred into a gamete. Why the somatic cell? Because the somatic cell is diploid, has a full set of chromosomes. It has the entire genome in it, chromosome pairs, okay? So it's the nucleus, it's the diploid nucleus of the somatic cell that is being transferred into an egg that has been enucleated. So that's essentially the process there. We take an egg, we take out its nucleus. So now we have an enucleated egg, and we replace that nucleus with a somatic nucleus. Functionally, we are generating a zygote, in other words, an egg with a diploid nucleus. All right, so functionally, it's like generating, like creating, manufacturing a zygote in the lab without reproduction, without fertilization. But the end result is an egg cytoplasm, an egg bag, if you will, that has a diploid nucleus in it. All right, now here's the trick. That diploid nucleus is a differentiated nucleus because it comes from a differentiated cell, somatic cell, like a skin cell or something. So that's a differentiated nucleus. It turns out when that differentiated nucleus is put inside the environment of a gamete, it undifferentiates. It differentiates back to the pluripotent stage and then moves forward into embryonic development to full differentiation. And that was Dolly. That was Dolly. Okay? So let's look for a moment at Dolly. And that's why I showed this slide here. This is the actual process. Here is an egg. You can still see the zona on it, all right, being held by the little vacuum. And here in this pipette, this glass pipette, here goes the nucleus. This is the somatic nucleus of a cell. Let's say it comes from the same female so that there's there's immune compatibility or genetic compatibility. But this is the nucleus from, let's say, a skin cell of this woman who donated this egg. This egg has been enucleated prior, right? So the pronucleus of the egg has been taken out, excised, so that there's no conflict. We don't want a trisomy here. We want just diploid chromosomes, all right? So what this does is it turns this egg cytoplasm into a zygote, because now this egg cytoplasm has within it a diploid nucleus, which is by definition, the functional definition of a zygote is a single cell with a diploid nucleus, okay? And that's what was done here. So the whole idea was to prove the principle that a mammal could be cloned, all right? So here is the sheep that is going to donate the nucleus, the diploid nucleus. It happens to come from the udder of the sheep, which is the mammary gland of the sheep, right? The skin. Huh? Yeah, the mammary gland, the udder, right? Udder is the mammary gland of cattle. And so here is the sheep, the female sheep who is donating the egg. This haploid nucleus, the pronucleus, is removed, right? So here's the egg cytoplasm. And here is the donor nucleus that is being inserted into the egg. In this case, it was done by an electrical shock. It could be by a pipette, a syringe glass pipette, or it can be done by electrical shock. There are several means of getting that nucleus in there. Now the nucleus is inside the egg. So now this is functionally a zygote because it has an egg cytoplasm and it has a diploid nucleus. Okay, so this functionally is a zygote who will then start embryonic development. And the next stage is the morula, which is here. The morula would actually have to grow to the blastocyst stage and then implant that blastocyst into a third sheep, all right? So that there were three, basically like three moms, if you will, for this sheep, for Dolly. There was the mom who donated the diploid nucleus. There was a different mom who donated the egg cytoplasm. And there was the gestational mom, okay? And a few months later, I don't know how much gestation takes on the sheep. Dolly was born. And the proof of principle was that Dolly grew to adult and had her own baby sheep. So she was fertile, okay? It's kind of a technicality, but we want to make sure that the cloned animal is fertile, okay? So that if she is infertile, then the infertility could be attributed to the technique, and therefore it's not a clean process. There's some defect happening because, you know, the cloned animal is sterile or infertile. But no, she was fertile.
So
Like-
since this is all
female?
Yeah.
Is the progeny is always female?
Not necessarily. Where is the sex determined on this clone? Is it on the gestational mom? She's just carrying the embryo, right? Is it on the egg cytoplasm? No, because that's just cytoplasm itself. Where is the X or XY gene? In the nucleus. So the nuclear donor will determine the sex of the clone. The nuclear donor, because that's where the X or the Y chromosome are going to be. So this could have been a male sheep. The male obviously doesn't have udder, right? So they have to obtain the cell from another part of the body.
Okay, so in this case, it would have to be female. But
Well, if they're going to use,
male,
yeah, it could have been a male.
Okay.
It could have been. I don't know why they used the udder. I don't know. You know, they happen to use the udder of the female, but they could have used another tissue, skin tissue from a male. I really don't know why the udder was chosen as a tissue of choice. Okay, so that's the process, and you can see that biologically there are 3 mothers associated with this clone, but it's a nuclear transfer, right? So the egg nucleus was taken out and was substituted by a somatic nucleus. And that somatic nucleus, by definition, is diploid and has the thing. But the interesting thing is that that somatic nucleus, in the environment of a egg cytoplasm, of a gamete cytoplasm, undifferentiates. So again, to Yamanaka, the factors are there. The developmental factors are there to undifferentiate that nucleus and then redifferentiate again. So one thing about nature is that nature is stubborn. You think we're stubborn. Nature is even more stubborn and tenacious. Nature is saying, "Oh, here I am, an egg with I'm an egg cytoplasm with a diploid nucleus. I better do what I'm supposed to do, go forward in differentiation and start the Morla process, etc., until the sheep came out a few months later or a few weeks later, whatever." Okay, that was done in '95 by Wilmot in Scotland in the Roslin Institute and Edinburgh. And there's Dolly, all right? Now, a little tongue in cheek. Why was she called a little side? Everybody know why she was called Dolly? Yes. To Wilmot's own words, all right? Because the key factor here is that diploid nucleus, in other words, the one that is containing the DNA, and since that diploid nucleus came from the udder of a sheep, the mammary glands, he said, I could not think of a more prodigious set of mammary glands than those of Dolly Parton's. So it's a tongue-in-cheek reference to Dolly Parton, who I happen to think she's a great actress and a very decent human being, but there it is. Okay, I don't know if he got sued out of his mind, but that's what happened. Okay, and that's why Dolly is named Dolly. Sorry? Yeah, it's still going on. In fact, it's now gone. So this was the first clone mammal. After that, there was a cat, and the cat was called Copycat. And then there was, yeah, Copycat, remember Copycat? And of course, the mouse always gets it. So now there are a lot of clone mice. And it's gone into animal husbandry for cattle to enhance. You know, you have a cow that's producing a lot of milk. Well, we're going to go after that udder. Right? Jacks?
It's a type of bovine cow.
Is it?
Mongolia or something like that.
Oh, yes? Oh.
Yeah, they used to clone them.
Yes?
The milk
is very strong.
It has more
Yak. Oh, yeah, the
Yeah.
yak. The yak, yes, from Mongolia. Right.
I think also cloning dogs. I think
Yes.
you can see people, rich people who have nothing better to do with their money.
Bingo. The pet industry has gotten into this. And the lady who lost her pet dog, you know, was her favorite friend, and now you can clone that dog and have another one like it. The clone was one of the hardest to clone. The dog was one of the hardest to clone. I read a few years ago, precisely because the female dog, her ovulation cycle is very difficult to detect when a female dog is actually ovulating. You know, and which, by the way, just in English grammar, for the record, a female dog technically is actually called a bitch, and that's why it's offensive to call a human a bitch, because it's referring to a female dog. And that's the origin of the word. Yeah, technically, officially, just like a lamb is called an ewe, a female dog is called a bitch. All right, just in science. And so it's very difficult to determine that bitch's ovulation. Speaking about a dog, and that's just the language. And that's why the dog was one of the most difficult ones to clone, because they couldn't determine when she was actually ovulating. But they finally got it, and now it's in the pet industry. And yes, the pets can be cloned that way, and there's money to be made because it's a market thing. Going to the human, then, what it will evolve for the human is to get a female to donate an egg, enucleate the egg, right? So now we got the cytoplasm of the egg, and then get a somatic cell, even from the same female or from another female or from a male, like for example, the husband, and generate that clone human. And now we have the egg with the somatic cell, the somatic nucleus. Sometimes they actually introduce the entire somatic cell, not just the nucleus. It's just easier. Instead of having to enucleate 2 cells, they just take the whole somatic cell and insert that cell into the cytoplasm, okay? Instead of having to enucleate the somatic cell also. It's just simply easier to insert the entire somatic cell. The cytoplasm of that somatic cell is going to be digested away. And then what's left is the nucleus, which would then proceed to the moral stage, blastocyst stage, in an incubator, and then implant or attempt to implant the baby, the blastocyst. 9 months later, then there would be the baby resulting. So this would be reproductive cloning. And this baby could have potentially 3 moms, right? The egg mom, the nucleus mom, and then the gestational mom. This baby could actually have a 4th mom, which is the one who is paying for all this and not donating any of the cells or womb. There could be a 4th mom who actually receives the baby 9 months after the gestation. So there could be a 4th mom here. As far as dads, it could be, well, this could be a dad instead of a mom. In other words, this could be a man donating the somatic nucleus, right? Could be a man instead of a woman. It's a somatic nucleus. So this could be the dad, but this dad would also be simultaneously the identical twin of that son. Okay, so the dad would be also the identical twin of that son because it's his somatic nucleus. The only difference is they may be 20 or 30 years apart in age, but they're identical twins. So now I'm the baby and my dad is also my identical twin. So again, the awkwardness, the bottom line, the manipulation of human life for just getting that baby from a clone, all right? And of course, the trials. I haven't even mentioned the trial and error in this because everybody knows about Dolly the sheep. A few people know that it took about 258 trials to get there, okay? And when they're doing other mammalian cloning, it's taking about 200 trials to get. So it seems to be something kind of hardwired in the mammals that the technology is taking about 200 to 250 trials to finally get one that's viable, that actually survives the pregnancy process, okay? So a lot of trial and error built into this, which when we translate that to the level of the human, of course, those are human killings, right?
Question. So if the boy would be an identical twin of the father who was the donor,
Right.
or it'll be an identical twin of the mother who was the donor?
Well, so it all depends where the nucleus comes from. Wherever the nucleus comes from, that's going to be the twin because it's his or her DNA.
So one of those 3 mothers will be the identical child? Or no?
Yeah, the second donor here, this one, because she's the one's donating the somatic cell. See, this
So
is,
she will look exactly like
yeah,
the donor. Whoever the donor is what the baby
yes,
will look like.
it will be the same sex and the same DNA, right?
Yeah.
You could also crunch this down to one, because the same woman who's donating the egg could also be donating the cell, the nucleus, and could also be gestating the baby. So you can crunch all this down to a single woman, you know, and then there's no father, because she's also donating the nucleus. So there's no father. And her daughter, by definition, the child would be a female, okay, because she's coming from the nucleus of a woman. So the daughter will be simultaneously her daughter and her identical twin sister. Hmm? Yeah, it's an artificial type of parthenogenesis, right? Manufactured in the lab.
Theory or you're doing?
There was some attempt, but because of the trials, and okay, so this goes back to Dolly. Your question brings me back to Dolly. It turns out that Dolly was euthanized prematurely. Dolly was killed at the Roslin on purpose, and I think this is actually the dummy. They stuffed her. They killed her, and then they stuffed her, and she's in a museum there at the Roslin, you know, in a vitrine. And I think this is actually, I'm not sure, but I think this is actually the stuff already, the stuffed animal. Why was Dolly euthanized after going through all that? Wasn't that supremely cruel to kill her? She had to, because it was what do you call that? It was compassionate euthanization because she started developing rheumatoid arthritis and a type of lung cancer. Diseases that are associated with old age for the sheep. Mm-hmm. And so she was aging prematurely, bottom line. She was aging prematurely. It turns out that apparently, and this has been observed in other clone mammals, is that it's successful and they're able to reproduce and everything else, but they age prematurely. So it seems like what is happening here functionally is that when this somatic nucleus is undifferentiated, is reverted back to undifferentiated to pluripotency, then when it starts differentiating again, it differentiates a little too fast. And they become aged prematurely.
Well, that's one of the reasons they talk about when women are freezing their eggs, when they do it at a younger age, they may not want to have a child until their careers and their families or whatever is established, but they're not using eggs that are 40 years old. They're actually using eggs that were 20 years old or
Correct.
25 or whatever age they froze them at.
Exactly. So biologically, there's an advantage there. Okay, right. And so with cloning, it's kind of a general pattern that with clone mammals, they age prematurely, faster. And so she was euthanized for humane reasons. After all, the principle was already proved, right? And she is, at the end of the day, a lab animal. So she was euthanized for humane reasons. But that same community of researchers then said we should not do reproductive cloning because based on experience, likely these children are going to age prematurely. And maybe as young adults are going to start developing issues that belong to older people, like arthritis and so forth. So it's inhumane to do this to children because this reproductive cloning, these children, we may be unwittingly doing an injustice to them by the fact that the evidence is showing that they're going to be aging prematurely. And so this community of researchers spoke very strongly against reproductive cloning. But it's the same community that wants to do therapeutic cloning. Okay, and they were so successful at it that they got California, I think it was Proposition 72 or something like that. There was a proposition in California that they extracted, I think at that time, about 10, 15 years ago, $300 million from the state funds to do human cloning for therapeutic reasons. In other words, this back then was seen as a way of bypassing killing human embryos. Okay, so we're not going to go after the human embryos. We're not going to go after the blastocysts of the human embryos. We're going to, through cloning, through human cloning, we're going to create these cell lines, these blastocysts that we're going to use for cell lines, okay? So for therapeutic cloning, the desire was to manufacture these human embryos in the lab so as to obtain the cell lines. But they're not killing human embryos that were already manufactured, if you will, either through in vitro, which was the access that they had, okay? In other words, they wanted to do therapeutic cloning to bypass killing human embryos, supposedly.
And how would that be immoral if to have an embryo? I think that you have to have the two things: the purpose of creating human life, so that that zygote is going to end up into a human being with that potential. But when you put a diploid nucleus and you grow it as a cell line, you're guiding into something that is just a part of it. How would that be interpreted as an embryo if there was no fertilization, conception per se? It was just a placement of a cell nucleus, your own cell. It's just a medium because the empty egg could come a part of a person. It's just a proto.
Yes, it's the medium. It's the medium. It's the environment in which it's propitious for that.
How would that be immoral?
Immoral, exactly. And that was their argument. How could this be immoral? Because an egg is not a human being, all right? And eggs die every month by the numbers. And a somatic nucleus is not a human being either because we're shedding skin cells all the time, right? And so we're putting these two cells together. Why should the product have any ethical quality, all right? And that was the argument. The rub is this, that it's not just putting that somatic nucleus into an egg, all right, into a nucleated egg. De facto, what is happening is that once that somatic nucleus is in there, it's going to undifferentiate back to the pluripotent stage. So functionally, it's becoming a zygote. And the proof of that is that then it starts differentiating exactly like a zygote does. In other words, it goes to the mortar, the blastocyst, and by the way, it's not the zygote that they were after, it's the ICM. So they have to do the cloning first, incubate that clone an average of 3 to 5 days until the blastocyst stage, and then extract the ICM, which would be the embryo proper. In other words, if we left that ICM alone in its place in situ, and then implanted that ICM into a woman, that ICM would normally continue growth, just like it did with Dolly, gastrulation, fetal stage, and eventually Dolly would come out, you know. So the thing is that, in fact, by doing this, they have generated a new human being. Even though for some people it's hard to see it because this is totally manufactured in the lab, taking an enucleated egg and inserting a somatic cell is certainly an artificial process. But in fact, what was happening is they were generating human beings in the lab. They were generating human zygotes, okay? But it was sold to the people of California as an alternative to killing human embryos, to going after.
Whether there's an installment at that moment. And there wasn't. We had
very good.
About
Is there installment?
Of conception with these two protonucleuses were to merge into a zygote.
Yeah.
But that process hasn't happened.
Correct. There's no fertilization. It's analogous to a parthenogenesis, like this, but there's no fertilization. However, so the question is fertilization a prerequisite for ensoulment? Or is rather humanity a prerequisite for ensoulment? Remember, I have at least a couple of cases to use as analogous. The twin, right? For monozygotic twins, when that other twin separates, there's no fertilization, and we have another human being. So we have other examples in nature, naturally, where there is the production, the generation of another human being without fertilization. We can call it a type of asexual reproduction, you know, because it's a radical separation between the morlin, and now we have a new human being, and there's no question that identical twins exist and are alive, and let's assume that they have a soul, right? That they're not soulless. So there's a natural process that parallels that. Secondly, it's not even the question that they have a soul. The question is that they have the possibility of a soul. And it does get philosophical, but we have to go there. And it's the old argument of the hunter and the bush. I think I mentioned this argument once. We're hunters, we love hunting, can't wait to go out hunting, and so we're going hunting. I know you hate hunting, but just humor myself for a moment that we love hunting and we're going out hunting. Okay, so we're out there and there's movement behind the bush, okay, and we know that there's, what's your favorite hunting animal? I don't know, a rabbit, a deer, whatever. So there's movement in the bush, and that could be the rabbit that we're trying to hunt, all right? But I know that there are other hunters out there. So that movement in the bush could be a rabbit or could be another hunter. Do I shoot or do I not shoot? Would I wait until I find out if it's another hunter or if it's a rabbit? Or do I just shoot because there's a possibility of a rabbit? There's a possibility of a rabbit, but there's a possibility of a hunter. I could kill a rabbit or I could kill a human being. Will I resist shooting or will I shoot? On the principle of bioethics, I may not shoot because there's a possibility, there's a real likely possibility that there's another human being there, you know? So I'm not at liberty to shoot until I have certainty, reasonable certainty that that's a rabbit and not a human being. So that's why I say just a possibility that this could receive a soul from God. And I think that the possibility is very real. It's not pie in the sky. I'm not talking about extraterrestrials here. Functionally, because how do I know that there's a real possibility that this is a human being?
Is the medium, the circumstances are right.
Exactly. We see the end product. The end product, just like Dolly came out a few months later, you know, baby would come out a few months later too. So there exists a real grounded, this is evidence-based. I'm not making this up. This is evidence-based. This now functional zygote would move forward into the morula, the blastocyst, the gastrula, etc. And 9 months, a baby would be there, just like Dolly. Okay, so we have evidence that this could move forward to produce an actual viable human being, and therefore we have to grant it the benefit of the doubt. Sorry, grant him or her the benefit of the doubt. Okay, and so that's why we need to abstain. This is the case where, when in doubt, abstain because it involves the life or death of another human being, the real life or death of another human being. And therefore, on principle ethics, we would need to abstain. We would, just for the possibility that God could be forced into granting a soul to this new human being, to this zygote. Right. Reproductive cloning. So the therapeutic cloning was for the purpose of generating cell lines for all the mesenchymal cell lines for the different 200 lines of the human body. All right. So it's very promising and so forth. The challenge is that, in a sense, if reproductive cloning is reprehensible because we're manufacturing this life in the lab with all the injustices that it involves, the end here, at least the end here, is to generate a baby for this couple, right? But the end here is doubly tainted in the sense with therapeutic cloning because sure, the end hypothetically is to create the cell lines, but we are generating human embryos in the lab on purpose to be killed. So these human embryos that are being generated in the lab, they're not even for the purpose of them to live, but the purpose of them to die for their cells. So you can say that in therapeutic cloning, there's really a double injustice built into it, in that the human embryos that are being manufactured are manufactured precisely for their ICM, in other words, precisely for being killed for their ICM. And that's the double injustice there. And that's where researchers who were so, let's see, emphatically against reproductive cloning and emphatically in favor of therapeutic cloning did not necessarily do the philosophical and the anthropological reasoning of realizing that this is ethically twice as tainted as the reproductive, because at least the reproductive was for giving the baby life. But here in therapeutic, the killing of that embryo is part and parcel of the process.
But what would be the rationale of doing the human cloning?
Well, yeah, it's right. So consider, well, personalized medicine. Say this person does have ALS, all right? And they're able to get a somatic cell that is clean of ALS, and they're able to manufacture a cell line for this person who has ALS and get her cured.
No, that I understand. But then I'm saying about the reproductive cloning.
For the reproductive?
Yeah, what's the rationale of that? Do I have to have a copy of myself? Or
Yeah, well, since, no, the thing is this, okay, so science is also a work in progress. And in the 90s, because the principle was proved that we can clone a mammal, all right? Well, biologically, we're mammals, so we can also clone a human. So why not clone a human, all right? And it's an alternative. It's like an alternative in vitro process that can be used, so that if the couple, or it's a single person, for example, a single man could be paying for the process and donating his, so he'll get an identical twin to himself, right? It's just options. Again, in a pragmatistic mentality or worldview, this is another option for generating a human being. And we can think of a number of people who have enough money to pay for this.
Father, when you're talking about ALS, for example, how would, I mean, what I know about ALS is minimal, but how would
Me
you
too.
be able
To cure it through this?
well, to cure it, because it isn't just one cell. And
It's
how would you
systemic,
find-
yeah.
Because theoretically, if there's a genetic component like the Huntington's or ALS or something else, it would still be in the genetic code that you're using. And what would you
Yeah.
take out to get rid of the ALS?
Exactly. I confess to not know enough, like yourself, about ALS and the etiology of it, the process. But let's say that hypothetically, in the somatic cell, in the somatic nucleus, I can engineer the somatic nucleus, maybe with CRISPR, all right? So that now the somatic nucleus is engineered to generate factors that could block the receptors of the ALS mechanism, all right? It just allows the somatic nucleus to be super engineered, and then to create the cell line, generate the cell line that is that immortal cell line, is that super cell line, if you will, the silver bullet.
Which is, this is the second part of the process. Usually comes with the genetic engineering first.
Right.
What it seeks is the compatibility, surety of the compatibility. So you do the genetic engineering in cell type, that is used like the mammary gland, which is fairly undifferentiated as an adult cell. So you're not going to use a neuron.
Oh, so that's what they. Oh, so that's what they use the other.
Cell as the precursor of your nucleus. No, you want a nucleus at an adult state, readily available, but fairly undifferentiated.
So it's more plastic dynamic.
Normally have the mammary
Okay,
gland.
that makes more sense.
Is that, so you will grab those cells and you try to slice the genes that are defective, are not coding for properly for the protein, and you try
Do the
to
correction there.
insert viruses, you have to insert
Well, it would
the
probably
right
benefit your
sequence, and then,
offspring versus you.
right, and
Exactly.
that is what you use as your precursor of deploying
Right.
nucleus into that process for the cell line. So in theory, you will get a clean cell line that
Right.
way. There's no way that you're gonna reject that as a tissue when it's inserted, and you have the fixed copy. So it's a double one. And that's what they do.
Yes, genetic engineering. It would involve
Yeah,
genetic
yeah,
engineering.
just a compliment.
Right, so the idea is to come up with the lines, and again, this was very big in the 90s to the point that California was thinking, because at that time, yes, so we were under the W administration, George W. Bush, and he had put a ban, a federal ban, at least on federal money, the big money, for doing human embryonic stem cell research, all right, based on pro-life principles. And so then it came down to the state level. But states wanted to do this, and California, innovative on the West Coast, they wanted to move forward that research. And I think it was Proposition 72, but it was taken to the population. Nope. Propositions.
Put California cloning,
Oh, yeah.
that's where I just pulled it up.
Okay, good. Yeah, to this day, there is a, there's a California dog. It takes on human
Yeah,
cloning.
Wired is the
Yeah,
article.
yeah. Exactly. I remember it was in the 90s because this was all happening at that time very fast. It was, yeah. Absolutely. Exactly. Yeah, but I was looking at I was crafting a bill that would place 5-year moratorium efforts to human clones.
Is your money good?
Yeah, no, but there was a proposition. Yeah. Let me just, yeah, because they carved out $3 billion. Here we go. $3 billion at the time. Okay, now this was Proposition 71. Proposition 71. When that ban came in, yeah, from W. So the federal funds dried up for cloning, for human cloning, but states, a number of states wanted to do this. And so California was a trailblazer on this and went forward with this proposition that extracted $3 billion in state funding to establish the Stem Cell Research Institute at California, Caltech. It was all the different, it was a state California university system, all right? And to this day, this institute is still going on. So they're the ones who are pioneering this, that we're going to speak loudly against human cloning, talking about reproductive cloning. But when you read the bill, all right, and was presented to the people of California, they said, no, we're not going to do human cloning, referring to reproductive cloning. What we're going to do is somatic cell nuclear transfer. We're going to do nuclear transfer, referring to therapeutic cloning. Okay, but it was so deceptive that they're using the language in double speak. They're saying, no, we want to ban human cloning, referring to reproductive cloning because of the genetic diseases, because of the premature aging. But at the same time, what we want to do is we want to do somatic cell nuclear transfer to develop these cell lines for healing all the diseases that beset humanity, meaning therapeutic cloning. Okay, and that's why I put the SCNT, because they were using that language deceptively to try to say, no, this is not cloning, meaning this is not reproductive cloning. But it was, in fact, therapeutic cloning. They just weren't calling that. They were calling it nuclear transfer. And you can read the actual bill, the actual proposal, and that's how it's presented to the people. And the people bought it, hook, line, and sinker. And to this day, there's that appropriation in the state legislature budget for this institute, okay? They're hard put to come up with results because it's just not working. In the meantime, the adults are working, the manaca is working, and the cord blood is working. But that's basically what I have to say about stem cell research and cloning. So I think I've covered kind of the basics of the biology and the bioethics that we're back to the human embryo, right? And the manipulation and the killing of that embryo. So from the principal perspective, we have to stay hands-on, all right? Thanks again, you've been very patient. We've gone beyond the time. Unfortunately, I apologize, but there it is. Now, the adults are working. They're not the panacea that they were supposed to be. What I've heard more recently is from treatments of adult stem cells, especially for regenerating tissues, is that they work for a while and then they start tapering off and the arthritis come back and so forth. So they have to do a boost of stem cells, of autologous stem cells. But it's working to some degree, but for some reason, over years, they tend to peter out and the person is going back to that condition. So they have to do a boost of stem cells again. And that's kind of where it's at right now. So, all right, you were mentioning, I ran out of time, but doctor, what you were mentioning to me earlier in the break of the fibrinogen being used for, fibrin being used for regeneration of tissue, I think we can fit it into next lecture, which is going to be sort of experimental things, but on the positive side of the human embryo, fetal surgery and fetal medicine, all right, on the positive side. So I'm going to go there for next week, next Saturday, on the lecture, on the last lecture on the beginning of life issues. All right. Okay. Thanks again. And we'll meet again next Saturday. Turn this off here. I want to take a little more coffee on the way. There may be some left.